Hello. All right, it's 11 o'clock. We'll go ahead and get started. Thanks for showing up to our session, Advances in Coupling Regional Climate and Social-Ecological Models to Improve Climate-Ready Ecosystem Management. I'm John Ream. We have several co-conveners, Tyler Eddy, Camilla Novagio, Stephen Bograd, Phoebe Woodworth, Jeff Coates, Kirsten Holzman, Roger Griffiths, a range of NOAA folks and FishMep folks, but we have submissions from people outside of those organizations, which is fantastic. I'm excited for our lineup. Today, to kick off things, we're going to have Ryan Heneghan from Queensland University of Technology, new approaches to understand humanity's impact on the world's oceans. Thanks.
So before I start, so it's just my name up there, but the work I'm going to show, it's definitely not a one-man band. I'm immensely privileged to be showing results from the FishMep community. So yeah, we'll see some of those people as we go through. But I just wanted to acknowledge that. So there's two parts to my talk. So first of all, I'm going to give an introduction to FishMep, and this as well will set the scene for several of the talks in this session who are FishMep affiliated researchers. And then I'm going to go into two critical challenges for global impact assessment, so global human impact assessment. So broadly, those two challenges are global models are uncertain and ensemble projections are expensive. So let's start with an introduction to FishMep. So FishMep is the Fisheries and Marine Ecosystem Model Intercomparison Project, which was established in 2013, I think at an ECHO meeting. And since then it's grown to about over 50 people working in the marine ecosystem, marine modeling space. So that encompasses about a dozen global marine ecosystem modelers, of which I'm one of them, or global marine ecosystem models, and then a couple of dozen regional models, which we'll hear a bit more about throughout this session. So FishMep's goals, broadly, there's two. So first of all, we want to provide robust projections of human impacts on marine ecosystems. And the second is we want to actually improve our understanding of marine ecosystems through developing our models, testing hypotheses, and pointing out where more work needs to be done. So how FishMep works? FishMep, we can think about it as sort of the marine, that it's sandwiched between Earth system models. So FishMep takes environmental and biogeochemical inputs from Earth system models. So these are things like temperature, primary production, plankton concentrations, oxygen, sea ice, those sorts of things. We run them through the marine ecosystem models, and the marine ecosystem models will then give us outputs such as total consumer biomass, catch, landings, and I'm talking here about global models, but the regional models do similar things as well. And the FishMep models are focused primarily on, well, they are focused on higher trophic levels. So if we think higher trophic levels, we're talking mainly about fish, benthic organisms, demersal, that sort of thing. So the first large study that FishMep conducted was presented in this 2019 paper led by Heike Lotzer. And so this was like the first time where we ran the global models together with the CMIP5 Earth system model ensembles, and we looked at changes in global biomass across the ensembles under different climate change scenarios. And one of the headline results from this study was, for the first time, we were able to get a relationship between the change in global marine animal biomass per degree of warming. So we were able to see a fairly linear change that per 1 degree of warming above pre-industrial levels, that we'd see a 5% decline in global fish biomass. More recently, so this was with CMIP5 projections, so more recently, as CMIP6 rolled out, FishMep worked again and wanted to see what was the change in our projections with the new CMIP6 ensemble compared to CMIP5. And what we saw is that projections of global biomass decline were greater when the models were forced by CMIP6 compared to CMIP5. And the reason for that mainly was higher temperature sensitivities in the Earth system models. So these studies so far have just been looking at the impacts of climate change in isolation, so working on a pristine, unfished ocean. And so we know that this is a pretty artificial, like a massive false assumption. It allows us to look at the impacts of climate change, but if we want to assess human impacts in total, we really need to move beyond that and look at global fishing. And this is because the impacts of industrial fishing since the Industrial Revolution exceed present or future worst-case climate projections. So this was a paper I co-led with Ian Hatton in 2021, and we reconstructed the global ocean size spectrum all the way from bacteria to whales. So this is looking at the distribution of biomass from the smallest to the largest organisms. And what we found, pulling results together, is that fishing to present has actually changed this scaling. So we've made the global ocean size spectrum steeper than what it was. And that's basically by eating and killing and hunting everything greater than 10 grams. So we've scooped out about 60% of the biomass of organisms greater than 10 grams. So FishMIP, to move beyond this, to move beyond just assuming a pristine ocean, what we're working on now is resolving the impacts of global fishing. And the point of that is twofold. So, we actually want to, we want to resolve the impacts of fishing for to assess human impact projections and the interplay between climate change and fishing. But we also want to, start to assess our models together, so look at model skill assessment. And that sort of brings me to the first big challenge, is that models are uncertain. So from the LOTSA study, with models, there's a bunch of different sources of uncertainty. With the FishMIP model, when we run things together as an ensemble, what we're trying to get at is we're wanting to look at structural uncertainty across the ensemble. And structural uncertainty is just, if I build a model and someone else builds a model, well, all the modelers, we're building models to answer slightly different questions, and we make different assumptions about what drives marine ecosystem structure and function. And all of that translates through to our models say different things about the future of the marine ecosystem. Basically what we saw is that the climate scenario uncertainty, so between a low emissions and a high emissions scenario, if we look at these mean lines, so the solid lines, if we look at the distance between these two lines and then compare it with the delta between here, we can see that the spread of our model uncertainty, so what this is looking at all the different models, their projections for one RCP. So I think it's RCP 8.5. And so we can see the spread of model uncertainty is far greater than the spread of the climate scenario uncertainty. And if we look at this spatially, so this is looking at a global integral. Spatially, we see there's variability in the distribution of changes, and then also where models agree. So models agree more in certain areas than others. So that sort of led us to ask, so what's driving this uncertainty? So as I said before, FishMIP takes, so the first, in terms of structural uncertainty, what's driving uncertainty is the models will all use different input parameters. So not only that, but we also capture structural uncertainty in Earth system models. So we're downstream from the Earth system models have uncertainty, we capture that, and then we have our own uncertainty. So we sort of see this compounding of uncertainty. So that's the first source. The second as well is that is the structural uncertainty in the marine ecosystem models, which I've already talked about. So as a community, we wanted to sort of pull apart, sort of disentangle the drivers of this uncertainty, so try and see where is this coming from structurally in our models. And so this was a paper that came out in 2021 where we started to work on that, so to pull these things apart. And what we did in this paper, we had 8 of our global marine models, so that was most of our global models, and we ran them through a simulation protocol where basically, the models have different inputs, so temperature inputs, inputs for lower trophic levels, and then other things like oxygen and pH. That's a small subset of models include that. Mainly the models are driven by these two categories. And so what we wanted to do is we wanted to try and pull out what is the effect of temperature, and then in effect how temperature is resolved in our models. What is the effect of the lower trophic level, how we resolve lower trophic levels in those linkages. And so what we did here is, I just want to focus on these two. We would, our temperature variables we would allow to change with climate change, but then we would have no climate change in our lower trophic level variables. So they would just be assumed that there's no climate change in this universe affects lower trophic levels. For temperature change, for lower trophic level change, we're doing the opposite. So we're keeping temperatures at pre-industrial levels and then allowing lower trophic levels to be affected. And then climate scenario, which is just everything varies. And so what we found pretty quickly, the first thing, so with the lower trophic level change results, is that we could pretty quickly split our model ensemble into two categories. So the first was models that use plankton carbon and models that use net primary production to represent lower trophic levels. And so so these are, all these maps are the change in variables under an RCP 8.5 scenario. So for these models that use plankton carbon, they're using phytoplankton or zooplankton, or maybe both. For these models that use primary production, they're using primary production. And pretty quickly, I think you can see that the change in each of the models, so these are each of our 8 marine ecosystem models in the ensemble. The change in higher trophic level biomass mirrors the change in lower trophic level the variable used to represent lower trophic levels. So that was the first thing we found. But more interesting than that, we saw this lower trophic level resolution bleeding through into how models responded to temperature. So we could still split models in their temperature response broadly, whether they use plankton carbon or whether they use primary production. The models that use plankton carbon were less sensitive to changes in sea surface temperature. So remember here, we're only varying temperature, we're not varying lower trophic level variables. So the models that use plankton carbon had less sensitivity to temperature increase than the models that use primary production. And this, from like a disentangling perspective, this was good because it we started to think, so what are the, how can we explain what's going on here? And so our hypothesis to explain what's going on in terms of model structure is that, so the models that use plankton have a one-way or an offline coupling between the Earth system models and the higher trophic levels. So the models take plankton carbon biomass for the ESMs, which serves as food for small fish, which are food for big fish, and then there's predation mortality from big on small, but there's no predation mortality. There's not a two-way linkage between small fish and plankton carbon biomass. What that effectively means is that in the models, as temperature goes up, respiration increases. It's not as much of a problem for small fish because they can eat more because the plankton biomass is essentially non-limiting. So that was our hypothesis for why our models that use plankton had less of a temperature sensitivity. The models that use primary production don't resolve predator-prey relationships at all. What they're doing is you have net primary production at the bottom of your, that's sustaining your food chain, and then at a given size, how much the productivity at that size is determined by transfer efficiency and PPMR, so pretty broad macroecological scalings. But what happens is that primary production and temperature set a cap on how much respiration you can have at a certain size class. And so as things get warmer, per unit biomass is respiring more, and so biomass becomes more expensive, so biomass goes down for a given unit of primary production. So what we found from this study is that our trophic linkages between lower and higher trophic levels are a fairly critical driver of uncertainty in the global fish meat model ensemble. So models are sensitive to the choice of lower trophic level forcing, and we saw that the changes in higher trophic levels biomass mirror what's going on at lower trophic levels. But we also saw that, that the discrepancies in temperature sensitivities are at least partly caused by how models resolve linkages between lower and higher trophic levels. And so we looked a little bit at parameters as well, so like how temperature affects scale in the different models, and I'll just go back. Because one thing we thought is maybe the temperature scalings in these models, maybe how things scale is less severe than these models. But this model and I think this model, they have the exact same temperature scaling. It's just more the processes that they're representing are very different, or how they're representing the linkages between lower and higher trophic levels is very different. So, models are improving. So, all of this work is because we want to improve our models. We want to resolve fish. We want to move forward, but we need impact projections now. And we need global impact projections for like strategic management to see where we're going on a global scale, to see where different strategies of, say, climate overshoot or geoengineering or global fishing strategies or climate change scenarios, how they are going to affect things at a global scale. So how do we make the best of the tools that we have? And so this last bit of the talk is going to be a bit more conceptual about where we're going as a community to sort of address these questions. So 2 challenges to recap is that models are uncertain, and we've seen that now, and we've sort of got to, we understand a little bit more about what's driving that structurally, but also ensemble projections are expensive. And so, the things I'm proposing in this next part are totally they're not novel. So the climate modeling community is way ahead in this area. But in terms of global ensemble projections, there's things we can learn from them and apply in the marine modeling space to push things forward. So the first for uncertainty in models is that we need to move beyond model democracy. So model democracy is that we run all of our models together as an ensemble with the same forcings, and then we weight them. So we take the mean of all the models, so we weight them all equally. So this is sort of a schematic of what that looks like if we update our weights. So like moving, if you've got two models, a red model and a blue model. The black line is, say, some observations, so an observed time series. The red model is always higher, the blue model is a lot lower. If you take the mean, then you're going to be, maybe the blue model is given a bit more weight than it should be in this situation. But if we update our weights based on model skill, maybe then hopefully we can reduce our uncertainty in our projections. And the reason why we want to do that is, first of all, like improve our projections, obviously, but also there's still a lot of uncertainty that this sort of a method would allow us to better target. So what areas, even when we do this, even if we can update if we move beyond model democracy, where are our models still most uncertain? And so this would allow us to better target that research. So that's uncertainty. The second is that, models are expensive. So FishMIP, since being established in 2013, has run 3 protocols, and we're on the 4th now. And these protocols take months to years to execute. And so there's a lot of different reasons for that, and a lot of things that, there are a lot of really, really good reasons for that. But there's a lot of appetite for human impact assessment, much more than what the community can actually respond to. And so, this is a massive bottleneck. And so one of the reasons for this is that this whole pipeline is quite expensive, especially when we're talking about the marine ecosystem models individually take, on high performance computing, can take days to weeks to run protocols, and that's multiplied across many different modeling groups, and so we see this compounding price in terms of computation, in terms of expertise, in terms of time. But if we look at this pathway, we can sort of think about our models as functions. So Well, they are functions. So marine ecosystem models, what they're doing is we're mapping environmental and social inputs to ecosystem outputs. So the models are like mathematical functions, basically. So, if we're wanting, so say the boats model, if we want to project fish biomass, boats is just a function that's mapping net primary production, temperature, and your fishing to give you fish biomass. And our models are largely deterministic and quite well behaved. So a small change in inputs leads to a small change in outputs. And what that means then is that we can start to think about things like model emulation. And so what we're working on now is a FishMIP ensemble emulator. And what the emulator will do is that, so FishMIP has already run many, a lot of, we have hundreds of gigabytes of simulations looking at different different SSPs, different fishing scenarios, that's what we're working on now. So we already have a lot of data on how our models respond to different inputs. And so what that allows us to do is to calibrate an emulator. All an emulator is a statistical model of a model. So because our models are well behaved, we can build approximations of those models that, instead of taking days to weeks on high performance computing, could take seconds to minutes on a desktop computer. And so an emulator of the FishMIP ensemble would allow us to rapidly assess novel environmental and social inputs to novel human impact assessments. And so the benefits of this, the main benefit of this, there's two. So it is speed, but coming from the thing of speed is also equity. So, if you imagine that to run these models, we need multiple modeling groups. And we need high-performance computing equipment, so it's expensive. But an emulator would increase equity of human impact projections, because you'd be able to, if you ran the emulator on your assessment, you're able to do that relatively cheaply in terms of time and computational power. So a desktop computer in minutes. And so this as well, this idea, as I said, is not new. So the climate modeling community in the IPCC latest report, most of the chapters used climate emulators to look, to go beyond the expensive simulations that were run with the climate models. I should stress as well, the point here is not to replace the marine ecosystem models. The emulator will only be as good as the underlying models. So, the point of this is to, maximize the use of what we've done and what we have right now. And putting all of this together, what it would allow us to do as well is to start to think about how can we look at human adaptation at the global scale. So if we work to address uncertainty, moving beyond model democracy, if we can address the speed bottleneck as well, so with an emulator, this allows us to move beyond a narrow subset of scenarios to start to look at how climate change might drive changes in fishing, which would then mitigate or alleviate or exacerbate climate impacts. So if you remember what I said earlier, how the impacts of historical fishing far exceed worst case projections of climate change. What that means is that from a modeling perspective, there is a huge potential to explore how can fishing be altered at a large scale to alleviate the impacts of climate change, but also as well. If we wanted to do that, we could just switch fishing off, but we can't do that. So what we need to do is we want to alleviate the impacts of climate change, say, but also maximize global fishing sustainably. And so an uncertainty-constrained emulator would allow us to explore that space. Okay, I'll just leave that, but I'll finish there. So thank you.
We got a couple minutes for questions.
Thank you. That was a really interesting talk. Kelly Carney, University of Washington. My question is regarding the weighting of models in your decreasing democracy section. In a lot of the studies, they've shown that fitting to data in the current period is not necessarily an indicator of skill in the future, and in fact, the models that are tuned too closely to a specific region are often the ones that are not as skilled at extrapolating into novel conditions. Are you considering that when you're looking at this weighting of models?
Yeah, so like the weighting of models, like I make it look like it's, you know, that there's like a lot of data to already do this. So the challenge with global models is, yeah, it's something that we need to do, but yeah, we need to consider that. So, you know, past performance is not an indicator of future performance, but we also need to think carefully about how we measure model skill, as you say. So we should look at things like, so in the latest protocol, we're looking at things like fish catch at the LME or the EEZ scale, and how that compares. But the higher level things that we need to look at maybe is to go back to our models and dig into them. So start to think, you know, maybe start to think about how our models resolve different processes and how realistic that is. So there's different frameworks of looking at uncertainty, and especially for projecting in the future, you want to make sure that your models are right for the right reasons. So I think we would need to be, you know, just comparing against data, but we also need to then still be looking at the mechanisms of our models to make sure that they're coherent and that they make sense. And I think that would give us a bit more confidence to push them into the future if we're sure of that the underlying mechanisms are okay as well.
Hi, thanks for the really cool talk. I'm excited about the emulator as a climate change economist, that I want to use it. But my question is on, so you mentioned that you run all these models with pristine seas, no fish. But I have used some of these, and as you showed, like some of the output data is like actually landings and total catch and things like that. So I wonder, like, what exactly are the socioeconomics of these, the assumptions? Is this catching affecting stocks, or what are the instructions like to the people that is modeling this in terms of fishing? Thanks.
Yeah, so for looking at so for the current protocol, we're using historical reconstructions of effort at like the large marine ecosystem scale. So I should say the model outputs in terms of catch and landings, global models are looking at aggregate biomass, so not like species specific, generally not species or functional group specific. Maybe we can get to functional groups sometimes, but it's generally aggregate biomass. It's quite a coarse resolution. But so the data that we're using for the latest protocol is, yeah, historical reconstructions from 1850 up to now of fish catch, fishing effort in different at the LME scale, and so that's skill assessment. But moving forward, yeah, there's a lot of work going on with what's called OSP, so ocean scenario pathways, and so those OSP pathways are looking at like coherent pathways of fishing effort globally that align with the SSP, so the shared socioeconomic pathways. So we're sort of, in terms of future scenarios of change, we're aligning closely with the huge body of work that's already been done on the SSPs, try and make sure that that's coherent.
Yeah. That wraps up pretty much all the time we have for questions. Thanks again, Ryan.
All right, now what we're going to do is we had 23 different poster presentations in the session, and we wanted to give a little bit of time for those that were interested to present a quick overview of their posters, and so we invited that. We've got 6 poster presentations that we're going to ask those poster authors to come up and give a 2-minute overview. So the first one that's up is going to be Ryan Heneghan, and we'll just ask oh, that was right. I'm sorry. Yes, sorry, my notes, obviously. Jason Holt, yes, thanks.
Hi, thank you. I'm Jason Holt from the National Oceanography Centre, and this is really an advert for a new UN Decade project that has just started. This is called Future Coastal Ocean Climates. It's part of the Coast Predict programme. What this is aiming to do is to see whether we can better coordinate climate downscaling for coastal ocean modelling around the world, possibly moving towards a global coastal ocean model into comparison experiments, so something that might, for example, underpin FishMIP that we've just heard about. So we're in very early days. We had our first workshop in February in Liverpool, and you see on the right there is a whole range of the modelling decisions that kind of underpin this. And if I can go to a next slide. If I did do two. Aha. Which part? Stand up there, everybody can see you. Okay. Okay, so I'll probably taken up all my time now. The poster is up, and I can go and, if you want to talk to me about this, then I'll be at around lunchtime. The door is very much open to any involvement and participation in it. There are a couple of QR codes where you can get some more information about the whole programme. But I'm guessing that's probably about it. Okay.
You did better than I did with my notes, so it's fantastic. So next up, we have Paul Getty. Great. And I'll just put, I'll put a finger up when you've got 30 seconds.
Sure. Well, thank you very much. So I'm Paul Getty. I'm a postdoctoral fellow currently working at Wassermann in France. So the work we are currently leading is trying to project the future of two fish stocks in the context of climate change. So the idea behind the model we are building is that everything in fish, or more or less, is related closely to the environment, especially the physiology and the ecology. So if you want to get a sense of what could be the population states by the middle or the end of the century, you need to account for that link. And so that's what we are trying to do with a bioenergetic approach, where you simulate the full life cycle of the fish as the response to environmental forcings. So that's what we do with a DEB model for those that know that approach. And then we integrate that DEB model to simulate the individual response within an IBM to upscale up to the population. So the poster is all about that, how we did that model for two stocks, two sea bass stocks in the Northeast Atlantic, how we designed the model, how we designed the simulations, and how we tried to calibrate the model on historical series just to see if we could capture the past trends and get a sense of how good the model could be. And in the end, so what we are trying to do, and these results are quite preliminary, but we are trying to do some projection using some RCP, so the 4.5 and the 8.5, and some very simplistic fishing scenarios. So I'd like just to thank my co-authors and the fundings, and also thank the people that already took the opportunity to give me some feedback during the poster session. And I'll be happy to hear more about what you guys think. So yeah, I'm all ears about any feedback that you could have. Thank you.
And then any questions for the first two speakers? I know it's quick. All right. And please find the posters afterwards and feel free to ask questions. Next up is Kelly Kearney. And you'll have 2 minutes, and we'll give you a little hands up when you're through.
Okay. First, I'll note that I changed the title of my poster to more closely reflect the question that I'm actually asking, which is how does biogeochemical process... Sorry. How does biogeochemical process uncertainty impact estimates of living marine resources in the Bering Sea? So this project came out of the ACLIME project, the Alaska Climate Integrated Modeling Project, which I think you'll hear a little bit more about later in this session. The basic idea is very similar to the FishMIP program that we just saw, except adding an additional downscaling step in there. So a suite of models from the CMIP6 ensemble is downscaled for the Bering Sea region, and then metrics are extracted and used for a number of different fisheries models. The idea is to estimate both the magnitude and the uncertainty of various living marine resources for better adaptive management in the future. However, you'll note in this little figure that there's a lot of ensembles happening. We use multiple socioeconomic scenarios, multiple global models, multiple fisheries models, but there's only a single biological model being used for the downscaling step itself. And my hypothesis was that we were severely underestimating some of the uncertainty that's associated with that step and possibly compressing some of the uncertainty upstream as we feed it through a single model. So to try to put some numbers on this, I've been running a biochemical model intercomparison with the downscaling. To do this, I attach 3, soon to be 4, different ecosystem models to a single regional ocean modeling system, ocean and ice model. These models are all, in and of themselves, equally justifiable from a point of view. They're all published, they're all validated, but they all take very different approaches to exactly what they emphasize and whether they're regional or more global in their aspects. So I had designed this presentation expecting this before the poster session, but yeah, in my poster I've been presenting some of the very preliminary results, and the short answer is things are very different between these models, even at very basic things, like whether there's a fall bloom or not, and what the magnitude of that fall bloom is very model dependent. So I'd be happy to discuss some of these preliminary details, and I look forward to the conversations.
Thanks, Kelly. And next up, we have Olivier Maury. Great. There you go. Thanks.
So hi everyone, I'm Alaia Morel. I'm a postdoc from the University of Montpellier, and I'm here to tease you my poster about forecasting fish communities under climate change with evolution. So to start the teasing of this poster, let's go far from marine ecology. I mean, very far. Let's talk about some dinosaurs from the past that would come directly on our present world. What could happen to them? Maybe some fiction, but maybe if we think about an evolutionary perspective, maybe because of temperature, disease, of the absence of the prey, so it wouldn't be very well in our world. So this can seem absurd, but for now, when we do future projection in marine ecosystem model, the fish have mainly their trait fixed, so they are fish in the present projected in the future. Whereas they can change through evolution or phenotypic plasticity, and it can impact demography, trophic web, or fisher incomes, for example. So in my work, I have included evolution and phenotypic plasticity in a marine ecosystem model, the OSMOS model, and I have applied it to the North Sea, and I did projection until the end of the century under climate scenario, and my goal was to understand the impact of evolution on the future projection. And it impacts species composition, catches, and fish size, sorry. But I will not spoil you the results. If you want to know more, come see me, and come see my poster, S8P12. Thank you.
Fantastic. And then last up, we have Roger Griffiths.
Yeah, good morning, Roger Griffiths, NOAA Fisheries Service, based in Silver Spring, Maryland, Washington, D.C. area. My poster's a little information about this end-to-end decision support system that we're trying to build to help advance climate-ready resource management. And the goal is to try and embody everything we're talking about during these 7 days or 5 days and try and put it into an operational system that we can better provide the information to the decision makers. Of course, it's trying to address these urgent needs about what are the best management strategies for fisheries management, to where should the future protected areas be, and what would climate community adaptation look like. Of course, the goal is, of course, to provide advice across time scales and spatial scales. Thank you, Kirsten, for the beautiful graphics. Of course, today we're challenged, I think, like many countries, many institutions. On the left side today, no regular delivery of the robust forecasts and projections that we need. really no operational system to turn that into the advice we need. We have a powerful research effort, but not an operational advice decision support system. And also low ability to use it in planning, and it results in very perhaps poor planning for extreme events and other things. Our hope is to produce a system that can do this kind of thing on an operational basis, end-to-end, particularly addressing the 4 requirements: reliable delivery of robust ocean forecasts and projections, operational ability to turn that into advice, increased decision-maker capacity, and of course the targeted research and observations to make sure it's continuously innovating. This is the cartoon of what the system would look like if we were able to build it. You can imagine that, you know, their entire talks or days of talks just focusing on the left side, the regional ocean modeling, let alone transferring that to say what are the ecological consequences and what's the advice, and of course building the decision makers' capability to use that information and the research effort to support it. We hope that we would build teams in each region to actually operationalize the system, and that if it worked right, the better cartoon is something like this, where the forecasts and projections are influencing or being used to by these decision support teams to produce the ecological advice and the human dimension advice, and that's being used to then provide decision makers with better outlooks to prepare. So what do we hope to do? Build that national system of ocean modeling, operational production of the advice. We're hoping that it then enables us to have more responsive management systems, and that it continuously innovates and validates over time. And of course, the hope, as we all do, is that it increases resilience and adaptation. So thank you very much.
That's great. Thanks, everyone, for the nice overview of the posters. We do have a few minutes for questions, if anybody has any quick questions, either for the posters or for the first speaker, which was not a poster, if you want to follow up now that you've had some time to think. All right.
Okay, well, we got 3 minutes.
Oh, yeah, go ahead. Go, Phoebe.
I actually have a question for Ryan, if we can
Yeah.
ask him a question. So for the emulators, if you put in novel conditions that aren't in those training scenarios, can it handle things that are well outside those bounds, or does that break down that training relationship?
Yeah, to break down. Yeah, I mean, it's like a statistical model, so you shouldn't be going outside the bounds of the data you have. The marine ecosystem models have been run over like no, low, high climate change scenarios, and like no fishing, sustainable fishing, open access. So that's what's working on now. So there is still a pretty big space to explore. But yeah, moving outside of that range, if that occurred, we would need more simulations. Yeah.
Okay, great.
Thank you. It's to Ryan as well. So regarding the model emulator, I think for some of the ecosystem models, it would make sense to emulate. Like we've emulated our Atlantis model, but for some of the other ones, like Thermicer, like they're just so fast that it doesn't provide any benefit. And then I'm also wondering if you thought of, so one thing that we encountered is how many simulations do we actually need to be able to emulate? And it's not a question that we answered necessarily. We just decided we were going to do 1,000 because it seemed kind of the level that we could handle. So I'm wondering if you've thought about that and how you've handled that.
No. Yeah, it's the same. So I'm thinking about the emulator, I'm thinking about global models, but there are global models that are extremely fast. And I suppose what we would want to do there is more bring them into like it's not just model speed, it's also that you need to engage like a dozen modeling groups to run the model. So if there is a way of running them like running those small, like cheap models with the rest of the emulator. That's what we would do there, rather than going to the trouble of emulating something that doesn't need to be emulated. And then the second question, I can't remember actually, sorry. Oh, number of simulations. Yeah, so we haven't thought too much about it yet, so this work is just starting, but I don't think we're constrained by the number of simulations, so we already have we would have like millions and millions of simulations that have already been run. If you look at like breaking down by different environmental conditions, it's a reasonably well-mapped space across the marine ecosystem models. So I think it'd be more a question of how do we sample that space in a proper way so that we're capturing it. Otherwise, yeah, we probably have too much data than too little, actually.
Thanks for that. All right, our next speaker is Tyler Eddy. More or less fish in future oceans? The importance of scale for climate change projections.
Thank you. And you're witnessing the Mac to PC conversion right there. So thank you for reading the title, John. So first off, I want to acknowledge the wide cast of collaborators and co-authors on this work that is FishMIP. This is really a group enterprise. On the far left, we have the Scenarios Working Group at FAO that took place in 2019. We had our latest FishMIP workshop in St. John's this past fall, and this was a side event that we had at the IMBER Future Oceans meeting in Brest that took place, I guess that was also pre-COVID. So this idea has been already introduced, but I just want to touch on it again. So the MIP experience, or the model intercomparison experience, really is trying to put models through their paces, do comparisons among models, and under this IPCC framework, evaluate whether or not there's a high level of agreement, and also evaluate the amount of evidence that there is to support the different conclusions. So we've heard in Ryan's talk and in some of the poster presentations about CMIP. There's also the EasyMIP project that FishMIP sits within. So they do agriculture projections, groundwater projections, energy projections, and this is all sort of set up through the IPCC framework. So we've seen this before, but just to refresh in case you just came in, FishMIP basically works by using environmental projections from the CMIP project, so typically primary production and temperature. We also use some socioeconomic projections, some assumptions about what's happening with fishing. We use these as inputs within our ensemble of marine ecosystem models, and then we compare not only within FishMIP, but we do it in a standardized format so that we can also compare with other sectors in EasyMIP. And this has allowed for a paper that Julia Blanchard led where we're comparing agricultural production to fisheries production. And so we've heard from Ryan about some of the different results from the global models within FishMIP, but we also have an ensemble of regional models. So, these models are quite different in how they've been developed and the philosophies that they sort of embrace. So we have some size or age-based style models. They might group all of fish in the ocean into 3 size categories. We have food web models, so these are your classic sort of ecopath, Atlantis models that actually resolve the food web connections. There's also some species distribution type models, as well as hybrid models. So this really gives us a range of different philosophies and modeling approaches to compare within FishMIP to, again, look at these different sources of variation and try to understand what's driving the projections within the models. So this is a slide that Ryan showed a similar one to, looking at our latest results from CMIP 6 for our global ensemble of marine ecosystem models. And as Ryan alluded to, we saw stronger declines under the CMIP 6 simulations compared to CMIP 5 simulations for the global models. But again, just highlighting that depending on which ecosystem model you look at, within the FishMIP ensemble, you get quite different magnitudes of the decline. And on average, we found that there's about a 2.5% greater decline with the CMIP6 scenarios compared to CMIP5, and this is for the total consumer biomass at a global scale. But one of the things we also noticed within this comparison of CMIP5 to CMIP6, and now we're looking at the change in biomass between these two model simulation rounds. And one of the things that we pointed out in this paper is that the regional shifts in the direction of biomass changes highlight the continued and urgent need to reduce uncertainty in the projected responses of marine ecosystems to climate change to help support adaptation planning. So we have these big global simulations, but a lot of management actions take place at regional scales. So understanding this source of variability at the regional scale is important for decision makers. So one of the questions we've been grappling with in FishMIP almost since the beginning is, do regional FishMIP models agree with the future projections for CMIP5 and CMIP6 simulations compared to this global model ensemble? And so highlighting different regions. So for example, if you're in Newfoundland, Canada, and you don't have necessarily a regional ecosystem model to look at the projections, can we take the output from these global model ensemble and use it at that regional scale? Oh, sorry for the formatting error, but so here we're looking at consumer biomass. This is under the CMIP5 projections. So we're looking at a comparison between a regional model for the Cook Strait of New Zealand compared to the subset from the global model ensemble within FishMIP. And so here we're looking at changes in consumer biomass. On the left, we have the GFDL simulations. On the right, we have the IPSL simulations. This is under an 8.5 scenario. And the green lines are the projections for the regional model, which is an ecopath with ecosim model. And then the red line is the multi-model mean, showing the variability in blue for the corresponding FishMIP global ensemble. And so one of the first things we started noticing within FishMIP is, in general, the global models sort of all agree on direction of change, usually a decline at the global scale. There is a fair amount of variability in the magnitude of decline, but we wanted to compare them to the regional models, and we hoped that that would sort of bolster, we'd have a little bit more confidence at the regional scale. And unfortunately, we often had one going up and the other going down. So this sort of forced us to go back to the drawing board, and as Ryan said, actually start to try to figure out what are the different mechanisms that are driving this variability. So when we did a bit more of a systematic comparison for all of the different regions where we had regional models, so we have Adriatic Sea, Baltic Sea, Cook Strait, East Bay Strait in Australia, so those are all ecopath models. We have the Humboldt Current, which is an osmos model, which you're going to hear later from Ricardo about, North Sea, Northwest Mediterranean, both ecopath, and then Southeast Australia, which is Beth's Atlantis model. So we compared the direction of change from the regional model to the global models, and we only found agreement in a couple of these different case studies. So we've been bashing this around for a number of years, trying to figure out, what are the reasons. Kelly presented a downscaled version of the environmental forcings, which I think, that's obviously the gold standard if you can have those downscaled ROMS models. A lot of regions in the world don't have that luxury, so we're sort of forced to work with what we've got. So we have since done another model simulation round with CMIP6, and we don't have all of the original models from the CMIP5 simulation that have repeated, and we have a number of new regions that have joined on. But one thing that we've noticed, now we're comparing the agreement between the global models and the regional models for the CMIP6. So where we do have these simulations, the overlap, we are starting to see that now, in this case, we have better agreement among the regional models and the global models. So this, again, confounds the story, because before we said they don't agree, and now we're saying perhaps they do agree under CMIP6 to more of an extent than they do in CMIP5. And again, with the caveat that we don't have the exact same sample set, for both of these different simulation rounds. So going back to the question, do regional and global FishMIP models agree on future projections for CMIP6 and CMIP6 simulations? So they mostly don't agree in CMIP 5, but they mostly do agree in CMIP 6. And again, you know, throughout FishMIP, we've been trying to understand how these models work and why they might project different things. So some of the different reasons that we've come up with, explanations. Regional models typically have greater functional diversity and ecological or taxonomic resolution. So typically regional models are developed to answer, you know, fishery-specific questions. So usually individual species that are relevant for fisheries are resolved within these regional models, where a lot of the global models sort of abstract to, you know, size classes. So they're kind of designed in different ways. The regional models generally include more processes and resolve predator-prey interactions more explicitly than global models. So perhaps there are different energy pathways that can mediate, you know, declines in phytoplankton groups, for example. They can, you know, energy can flow through the food web in different ways if you have resolved these predator-prey interactions. And as something that's been brought up before, the core spatial resolution of coastal regions in global Earth system models and FishMIP models, while the regional FishMIP models are developed at finer scales. And this has also been a challenge that we've had to face, is how do we deal with global Earth system models developed at a 1 degree grid, which is about 100 kilometer scale, that might be missing, you know, coastal primary productivity that we know is really important and that a lot of regional models have been calibrated to. So we had to come up with some workarounds with that, but that's something that we're developing or we're addressing in the present simulation round as well. So I'm just going to sum up here. Variation in model projections is a good thing. I'll try to convince you of that. It can teach us things about our models. It can be frustrating at times, but hopefully we can use it as a learning experience. Spatial resolution is an important factor to consider for climate change projections using marine ecosystem models. And I know there are a lot of studies that try to use global projections at regional scale, so I just, you know, put this caution here. You know, there is this mismatch in some case if you are looking at coastal systems. The first question might be is whether the Earth system model is actually capturing that, you know, coastal variation. And our present FishMIP simulation round is poised to tease out the contribution of spatial scale to variation in regional and global model projections. So one of our simulations is run at the 1 degree grid scale, and we have a equivalent simulation run at the 1/4 degree grid scale. So it's not as fine scale as some of these ROMS models get at, but we're interested to see if that makes a big difference in explaining variation. And we heard the other day, I think during one of the keynotes on Tuesday morning, to be hard on your models. I agree, but also be nice to your fellow modelers. And that's why FishMIP's been so successful, so I'd like to thank you guys.
Tyler, we got time for a question or two. Paul, I saw your hand go up first.
Oh, yeah. So it seems like sometimes you can get cases where two models might agree in the kind of the direction of change, but there could be really drastic differences in the rates of change and the scale. Mm-hmm. Have you noticed that, or have you thought about kind of more refined criteria for what agreement is?
Among the global models, or regional, or both?
Well,
or?
it kind of in a comparison between, for both, comparison between the regional models and the global models.
Yes, we have looked within the global ensemble, and it seemed that some of the more size-based global models projected the stronger declines, whereas some of the models that included some of these trophic interactions had smaller declines. So that's sort of a general observation. There are a few exception models within FishMIP, but as always, but that's sort of a general observation.
All right, one last question. Is it brief?
The brief version is, okay, so with the ecopath models, usually there's not one standard way of incorporating, you know, temperature or climate. Do you know if the models that you were talking about all incorporated it in the same way, or if that was variable? Yes
yeah.
Or no?
Yes, with the asterisks.
Okay.
All right, thank you. All right, our next speaker, Jerome Guiet. He's abroad, he's online. Oh, right. So we're going to be playing a video, right? Okay.
Hi everyone, and thank you to the organizers for giving me the opportunity to talk this session today. Unfortunately, I could not attend this conference in person, but I am available online if you have questions or feedbacks. So the title of this talk, Inconsistent Fishing Effort Forcing Can Lead to Unrealistically Optimistic Future Projections. I am presenting today, but this work includes the contribution of other people, Daniel Bianchi, Kim Scherer, and Eric Galbraith. Okay, so first, about the context. Global fish biomass projections, especially from the FishMIP intercomparison project, suggest a decline of the fish biomass in a changing ocean. So that's what is illustrated here with this figure from the paper of Direct Intensor that shows that for different climatic scenarios, the fish biomass declines at different rates with some substantial variability between across scenarios. It's not surprising since it includes different models which have very different structure, different forcing variables, or different sensitivities. So since these simulations, a new round of simulation with FishMIP is in preparation, which will partly address or attempt to address this uncertainty, simulating the historical catches when models are forced with observed climate and observed fishing effort. Along this simulation, we started to wonder what will be the implication in our model, both of forcing both climate and fishing effort, as it can lead to inconsistencies of where the biomass is produced and where the fishing effort is applied. I will explain this more in the coming slides. But first, about the BOSS model, just for those who don't know, in a nutshell, this model reproduces the biomass distribution from few environmental forcings, which are the primary production and the temperature. So basically, this primary production will provide food for organisms, and temperature will affect metabolic rates. Ultimately, determining the dynamic of this distribution as a function of time, and such kind of distributions are in each grid cells of our domain. And what you can see, for example, like different biomasses when everything is summed together. So in its initial formulation, BOSS is also coupled with a dynamic fishing effort modules, which relies on open access assumptions. So just in few words, from this fish biomass distribution, the model will be able to access a fraction, which we call accessible biomass. And then, using an elementary economic dynamics, the model, assuming an exponential increase of catchability, reproduce the development of fisheries. So just in few, let me explain you. In each grid cell of the domain, the model will start from the preceding biomasses, predetermined at this part here, and then as catchability increase, at some point this biomass becomes profitable to exploit, efforts start increasing, catches as well. Biomass is then progressively depleted up to a point where there is so little biomass that catches are not increasing anymore, so they reach a peak, and eventually as effort keep increasing in the future, the nominal effort will at this turn start decreasing. And the point with such a formulation is that there is a connection between the biomass production, the effort development, and the catches. And now, with this issue of forcing effort, one can wonder how breaking these connections will impact biomasses and catches, and ultimately simulations and projections of the fisheries. So in order to test this, we conducted 4 sets of experiments. First, the open access simulation I was just presenting. So basically, in such a formulation, in each region, say large marine ecosystems, there is some kind of relationship between the pristine biomass and the total effort that will be applied, while within each region as well, there is, on the other side, a lack of connection between the biomass and effort at the same time step, simply because in different grid cells, you will have different rates of development of fisheries. So that's our reference simulation that I will first compare with what I call open access bio, which is basically a simulation which takes the same effort at this open access run here, average it at each time step in each LME, and redistribute it proportionally to biomass. So the difference between these two is here that instead of having a disconnect at each time step between effort and biomass, we now have a proportionality between these two. Third simulation that I called OBS bio. Now we increasingly disconnect the regions where the biomass is produced and the regions where effort is applied, redistributing the effort observed here to match spatial patterns that are observed actually. So in this third run, now the biomass, the pristine biomass in each large marine ecosystem will not match exactly the effort which is applied in each region. There's still a relationship between the biomass and effort at each time step, because again, like this mean effort in each region will be distributed proportionally to the biomass. And finally, the most extreme case, from this one, we now redistribute the effort in each region not proportionally to biomass, but using observed gridded effort distribution. So that's the last step. Now, at each time step, there is not only a difference between where the biomass and effort within the LME are, there's not only a mismatch here, there's also a mismatch at each time step between where the biomass is and effort in each region. So from here to here, you have somewhat of a gradient of moving toward the more observed fishing effort, but you have also a gradient from an increased error of where the effort is applied and where the biomass will normally dynamically expect effort to develop. Okay, so we'll compare these two simulations first, and that's what you see here. So all these runs have overall the same nominal effort across LMEs, but now it's more about how this effort is distributed in space. So moving from the open access to a case where within each LME the biomass is distributed, the effort is distributed to biomass, we first observed an increase of the remaining biomass across time that will also be linked to higher maximum catches within all large marine ecosystems. So that's the first point here, this first disconnect between biomass and effort. Distributing effort proportionally to biomass results in comparatively higher biomass and will maximize yields. Now, moving to our more observed effort, when I redistribute effort between LMEs to match observations, we have this second situation. Again, same nominal effort across time, but now instead of having a mismatch in the biomass at the beginning, before the year 2000, it's in the future that we will have larger biomass remainings that will then hold slightly larger catches. What's happening here? We presume that as effort is redistributed in space between the two simulations, the underexploited regions will stay longer, maintaining higher biomasses that will hold larger catches. And the second message here is that mismatching biomass from effort at a large marine ecosystem level results in remaining biomasses that support larger catches in the future. And as we now increase the mismatch effort biomass, moving toward the last simulation I just described, again, more and more biomass remain. It starts to be a substantial difference between different runs that will also have like strong impact on the future catch potential in the modeled in the simulations. Now, as a side note, obviously the open access dynamic I was talking about is a bit of an extreme case, since management is already affecting the way effort is distributing across regions. But the point here is that these different runs reveal that larger biomasses remain when we force effort compared to a fully dynamic case, and these differences in remaining biomasses will lead to different responses in ecosystems to, say, different effort scenarios. As an example here, when I fix, for example, effort at the 2010 level, you can see that for these two simulations, substantial difference in remaining biomass occurs that will affect the catch potential or the projections. We have here, for example, a difference of 15%. Okay, in summary, and addressing this question, what are the implications of inconsistent effort forcing? The take-home message is that as effort is forced, increasingly independently of where the model produces biomass, we end up having less depletion of ecosystems. And as you can see, we keep larger and larger biomasses in these ecosystems that ultimately will impact catches, time series, but also, and especially the future catches, but also the response of these ecosystems to different management or regulations of effort. This is results based on the bots model, but it seemed relevant to us to make these points as this might affect other models, and that might be something to keep in mind, especially as today I was just talking about this spatial mismatch of effort, but inconsistencies are not only in space, but also depending on how we force effort between fish size classes or different functional types. Thank you for your attention, and if you have questions, I am, as I said, available online to answer them. Thank you very much.
By chance, are any of Jerome's collaborators in the audience? Okay, all right, so maybe you can field questions. Yeah, we got about 3 minutes. Any questions? Yeah. No? Do you want to tell us your feelings about all that?
Ooh, I was not prepared, so no.
Okay, that's fine. We can also.
Like, is this something? Yeah. I think it's. I don't know what the other modelers think. Like, if you have biomass that's distributed in one way because of the model and the forcing, and then you force it with effort that's different. Any like? Do you think it might be a problem?
I think the results are consistent with what we're seeing in terms of different harvest rules that take into account whether or not there is full exploitation or that one-to-one effort with biomass. So I found it consistent with what we're finding with the different methodology.
3 minutes.
Yeah. We can also, questions, we can open them back up for Tyler, if anyone has any. Right. But you can always email Jerome.
All right, we'll head on to our next speaker, Jeroen Steenbeek, who will be guiding us on a systematic exploration of ecological responses to divergent climate trajectories and initialized Earth system predictions.
Hello, everybody. Nice. Thanks. Honored to be here. What I'm going to present to you is something somewhat different. My background is not in ecology. My background is in software engineering. And I started my PhD here in the top back in 2000, a month before COVID erupted, on the subject on are ecosystem models ready to face the challenges raised by the ocean decade. And this exercise is in that light. So just to reiterate a little bit how I see the world from my developer's point of view, we're talking about marine megatistic ecosystem models, marine ecosystem models, that represent whole food webs over time and space. You can look at the past to understand what happened, and then you can extrapolate in the future with some level of certainty, many times used word here, in order to see what the future might hold. And these models are increasingly used for policy management, for cumulative impact assessments and all that. There are a different breed of models. I think it's my beard you're hearing, sorry. Anyway. So these models are complex, iterative, and as such, very clumsy to run. They're a different type of breed of model that is not easily systematically assessed. And that's indeed the issue we find here. If you look overall, marine ecosystem models are not often validated, systematically calibrated, or assessed for uncertainty. We've seen a few cases here that do it. It's fantastic. But if you look at the literature, this is a very big underrepresented world, and that is needed. There are several reasons for it. We wrote a position paper in 2021. Here's the reference. Yeah, I needed to shave, I'm sorry. Anyway. Good. With that out of the way, why are marine ecosystem models rarely assessed? The literature explodes with suggestions for skill metrics, frameworks, methodologies to do it, but it's not being done. So the ecosystem modeling community struggles to put theory to practice. And this sentence here, it's basically 4 separate sentences that are all connected. There is in academia a lack of funding, like funding in general, either to re-engineer models. Science is always focused on producing new science. You're not going to re-engineer something that already existed, although in this case, that's needed, right? That's a bottleneck. You can't really evolve IT expertise that way because the salaries are not competitive in order to help you further. MEMS, marine ecosystem models, have a legacy structure that makes them just behemoths to run. You cannot split them up. You cannot just farm them out in little pieces. It doesn't work. And then you have to have capable hardware. So these 4 factors together create a bit of a conundrum, create a problem. So what we thought, we were dreaming up a system. It's basically represented here. We have your MEM development and you've got all the theory, but there's this big technological gap that prevents us from doing what we should be doing. Also because it's probably not really that mandated, even in review processes. So anyway, it wouldn't be great, it wouldn't be great if we had a simple and practical solution to cross this gap as a scaffolding for doing the better exercises. Well, then I got involved in the Atlas project, who is basically run from the university in Bergen. And they focus on the predictability of Earth system model and marine ecosystem model complexes, and especially on the short time scale, seasonal, decadal, seasonal, annual, biannual to decadal time scales. It's a gap where, it's nice to have long-term projections, but the most decision space is in the shorter time span. And the marine ecosystem models are quite bad at performing there. And the Earth system modeling world has moved far ahead of it. The Earth system modeling world is older. Several reasons for this. But anyway, so under Atlas, the aim is to assess this. Can we do that? So this is basically what we want to do in this case study I'm presenting here. So if you look at an historical period, where marine Earth system models do, they have these different members, the different realizations that start annually to feather out and to see, okay, based on a historical baseline, what kind of spread do we get in the results? Our thought was just, okay, what if we pump this kind of stuff, like hundreds of runs, through a marine ecosystem model, what happens? So it's basically, banging the rocks together and see what happened. And see if we can build an infrastructure that can do this, because we can't. Not most of us. So that's the challenge that we raised ourselves. In that position paper, we conceptualized a bit what a management framework would look like, and what you actually want. If you don't have access to big funding, you don't have IT staff to support yourself, you cannot re-engineer a marine ecosystem model, what would you do? So what you ideally would like to do is simply have a desktop process where you tie a few scripts together, a bit of R, marine ecosystem model, maybe some Python, maybe through MATLAB, whatever your choice tools are. If you can do this systematically on one computer, on multiple computers or a cluster, then we have something. So I set out to build something like this, just to see if you could do this. It has to be as simple to deploy as a KISS. Keep it simple, stupid. That's an acronym. You just try to make it as simple as possible without reliance on anything that is expensive, inaccessible, or requires other people to help you run it. It's quite an ambitious task, but okay, it should be doable. So the next slide, it's going to be rough. This is what I built. I'm not going to explain this whole thing. Take a picture and mull over it at home if you want, or watch the recordings. But what this basically says is there's 4 separate layers that are totally independent, where your science lies somewhere in the middle. This is what you really care about. The rest is peripheral. So on a given hardware, I have built a framework that just listens to what the science wants to do and farms out a workload across a series of available clients where the tasks get done. And it could be on the same computer, could be on a computing cluster, could be across the internet, could be anywhere. The system does not constrain you in any way. Just simply sequentially if your workload with several jobs go to different clients, and the job consists of a little bit of R, a little bit of marine ecosystem model, that, and the results get put back. And when this whole workflow is done, in the end, the controlling application gets the last shot at it. I'm done, these are my results. Do you want to post a new workload or you want to stop? So this allows you to basically iterate. You can basically build anything on this. That's the idea about this framework. And I've tested this with all these languages in parallel, passing a bit of code, a bit of data through R, through Python, through Perl, through Java, etc., just to show, yeah, it can be done. Okay, but now you need the infrastructure to do this, right? So we built this monster. We tested it in a case study where we have the Earth system model output from two Earth system models that work with us on the Triatlas. We have the North CPM model built here in Bergen and the EC Earth model in the Barcelona supercomputing group. And they delivered, we only decided to look at 3 different members through different per annual feathers, if you want to call it. And they started annually in November 1980. We pumped this to Poor Eco Ocean, a global ecosystem footprint model that consists of 51 functional groups, but represents 3,400 species, accounting for species diversity, different responses, physiology, etc. And also things like temperature, adjusted metabolic rates, and all these things to capture the global footprint. And Eco Ocean participates in FishMap. And we drive this via the Earth system model output. Only using at the phytoplankton concentrations and temperature, but just as a proof of concept, to see if you could do this. This is how we execute the Eco-Ocean, and the cool thing is this were 384 runs that we passed through the system, looking at fish and non-fish oceans, and we really run it on a wild agglomeration of computers, 2 desktops and a couple of laptops in 2 different locations, just to see, okay, could you do this, right? The coolest thing is that these tools were linked through Sync and Dropbox to cloud providers. It had to be really low tech. That was the self-imposed limit. We use Sync for the communication of framework. So the server says, I want to run this. Client, oh, that's for me. I'll read it. And so it's just really file-based transfer for Dropbox and Sync. We started off with one provider, but we realized that the big data that came out of the model stifled the framework communication, so we split that. 2 low-key tools that everybody can set up. The cool thing about, of course, Dropbox and Sync is you just point at a folder and it works, right? You don't have to pass firewalls. You don't have IT. You don't need to do anything. That's complicated. And with this setup, to our surprise, all these simulations were done in 24 hours, but it consumed in 24 hours 1,600 hours of CPU time. That's insane. If you do this on Amazon or Google, your scientific budget is shot. You can't, but you can do it at home. And that is so cool about this. Anyway, so, and also an interesting thing is that, it produced a ton of output, but we did not bother for every run storing all the big maps that Eco-Ocean produces, but we just intercepted the data as time series across different regions in order to capture interesting signals, and then with the philosophy that later, if you want to post and analyze this, from the time series you can see where things go haywire, and we can easily rerun those runs again, rather than having to save 5 terabytes of output and sift through that. So it's an optimization. But this is indicative how I think you can use a framework like this. Don't do everything, do it smartly. So anyway, some initial results. You can see here, here above we have small pelagic, here's large pelagics. We have a hypothetical no-fished ocean that has, thank God, already been explained today, somewhere in this session. And we fished with horizontal fission. And you can see, I mean, if no fission, your large pelagics deplete your small pelagics. If you start fishing, then the trend changes. But you also see immediately that the feathers that you see here start flattening out, which is a scaling issue, which indicates that fishing is much more impactful. we have to dig deeper in. The runs are now completed, but we have to start analyzing this. But I think I wanted to present the framework, the ideas behind the framework, and we'll dig deeper in serious scientific papers next. So, but the next, I think the whole idea is promising, and we would like to, I would like to propose to FishMIP to develop this framework further, or at least the ideas within, because I think with FishMIP we have such a body of smart people together, we can set the standard for the rest of the ecosystem modeling world, and that's cool. Yeah, I think. Anyway, so the preliminary insights. Yeah, Earth system model variability does propagate to the food web. Doesn't seem to be as profound as fishing impacts, but there are some regions that behave differently, and those are the regions we really want to dig into, because that tells us much more about how the modeling process works. So it's still a very valuable exercise. And because we haven't sifted through literally the hundreds of thousands of time series we've gotten, that's another process. So that will come next. And I hope to tell you more interesting things about that later. There's some preliminary conclusions about the framework itself. Because you now suddenly have computing power, it doesn't mean you should go gung-ho. You should be careful what you run, in measure, and be clever, because otherwise it becomes unwielding. So there's this ever trade-off. The use of Dropbox and Sync, I don't recommend it. It was absolutely painful, but it worked, and that was the whole point. So, yeah, I think the potential of the framework is big. This is really, with no big ambitions, you can do really ambitious things, and I think that's cool. And this basically allows us now to start, as a stepping stone, to start modeling with a bit more confidence for the big things that we want to achieve. So that's cool. Nice. And with this, thank you. Nice. Do you know him? No, it's MacGyver. It's my hero.
We have time for questions. Do you have one?
Thanks, that was great. Are there thoughts, ideas about expanding this into a citizen science type approach? And you know, do you have a plan and a strategy for that, for getting everyone to run this on their computers?
Oh yeah, I can talk here. I don't have to get the microphone. That's true. This would be great. I mean, I think the potential for this is big. My thesis also focused on what was raised just with you, Ron, with John, about communication. So how can you bridge the gap between what the model is doing, what the audience needs, and bring it back? So those kind of things we work on as well. I mean, the applications are you could apply to anything. Stakeholders, yeah. I mean, this allows you. Well, if you unlock this kind of capabilities, you can basically run modeling sessions with stakeholders, but run them in parallel to, within a reasonable time, get an uncertainty spread. Something you couldn't do on a single laptop. If you bring a few and you have your Dropbox and Sync framework, all the clunkiness, you can still do it. So can you imagine if you scale this up to a real infrastructure, what you would get? So I think, yeah.
Are there other questions? I saw Cheryl.
I think this is really exciting, especially the part about what places are sensitive to fishing and which aren't. And we were talking earlier about which places or system models are well represented at 1 degree and where they aren't. So you could start to do like kind of this compounded uncertainty that could be given to management. And I think we'll talk more.
Nice.
It's really cool.
I think what you can do, I mean, this is only a small number of variations. With this kind of computing power, you can, of course, start varying systematically. We have an eco-ocean paper where you turn internal hypotheses on or off, just to see what matters. And you can start combining this. I mean, now the run length is not the issue anymore. The issue will be how to make sense of all the output. I don't want to touch that right now, but that's another issue. But the power is big. Oh yeah, we'll keep writing. Nice.
All right, is it a super quick question? Okay. Well, thanks again. That was really inspiring.
Nice. Thank you.
Okay, I think, is our next talk a recorded talk by Desiree Tommasi? Yes, okay.
Hi everyone, it's great to be here to talk about the Future Seas project. I'm the lead PI, and first off I would like to acknowledge the many co-authors listed here who contributed to the work I will be presenting. The aim of Future Seas is to explore potential impacts of climate change on US West Coast fisheries and to evaluate strategies for managing those impacts. We are focusing on the California current ecosystem along the US West Coast. This is a productive upwelling system that provides many ecosystem services, and the forage assemblage plays a key role transferring energy from plankton to upper trophic levels, including protected species and commercial and recreational fisheries. The project so far has focused on 3 key fisheries of interest: albacore tuna, swordfish, and coastal pelagic species. Today I will highlight results of work on coastal pelagic species and on Pacific sardine in particular. Here is an overview of our modeling approach. It first integrates the downscale projections of ocean conditions, also developed under Future Seas. Those inform species distribution models and a set of biological models ranging from the Atlantis whole ecosystem model to a single species sardine model. These biological models have different parameterizations and assumptions, and we use an ensemble to capture uncertainty in the response of our forage species of interest to climate change. We are also developing economic models to assess impacts on fishing communities and a management strategy evaluation to assess robustness of management measures to climate-driven changes in forage fish dynamics. The first step of the project consisted in using a ROMS model to downscale climate projections from 3 Earth system models, Hadley, GFDL, and IPSL. Our downscale climate projections show that without strong curbing of emissions, the California current will undergo significant change this century, including a 2 to 4 degree warming in SST. If you want to learn more about the downscaling process and patterns in other variables, please check out the paper by Merpoz-Obuil that's linked here. We also show that by the end of century, there will be an almost ubiquitous shift to novel conditions. These maps show an assessment of the novelty of climate envelopes defined by temperature, dissolved oxygen, mixed layer depth, and eddy kinetic energy for August at the end of the 5 decades represented here under ROMS IPSL. In 2020, the map is green, showing analog conditions. However, by the end of century, most of the map is red, showing conditions that have never historically been experienced in the California current. We were then interested in assessing the impact of those physical and biogeochemical changes on the distribution of our species of interest. Here, I show the results for the northern subpopulation of Pacific sardine. On the left, for each of the ESMs, we show the difference in sardine habitat suitability for March between 2040 to 2055 and 2000 to 2015. So the purple color signifies an increase in habitat suitability in 2040 to 2055 versus the historical period. Habitat suitability here was derived from a statistical species distribution model developed by Barbara Muning. On the right, we show maps of distribution of adult sardine at the top and sardine eggs at the bottom from a mechanistic individual-based model that was developed by Jerome Fichter. In the model, movement of adults and juveniles follows behavioral cues, whereas movement of eggs and larvae is driven by ocean currents. Both models show a poleward shift in the habitat of the sardine northern subpopulation. Not only are we interested in how the projected oceanographic changes will affect species distribution, but also their productivity and abundance. Here are the results of population projections for sardine developed with a mechanistic age structure model which includes impacts of both food and temperature on early life stages survival. You can see that projections of future sardine population differ by climate model, and so they're more uncertain than the species distribution projection that I've shown before. There is also large uncertainty associated with the response to temperature that is outside what was observed in the historical period, and that's represented by the gray shading over here, the thermal response uncertainty. If you want to learn more about the model, please read the paper by Stefan Koenigstein. He's the developer of this model, and also check out his poster in this session. These projected changes in species dynamics lead to emerging management needs brought on by climate change. In our region, managers and stakeholders want to understand how shifting habitats will affect survey planning, stock structure, transboundary management issues, bycatch issues, changing prey interactions, changing social vulnerability, as well as emerging fisheries. In the following slides, I briefly provide an overview of how what Future Seas has learned in terms of future sardine distribution is going to affect survey planning, stock structure, transboundary management, as well as changing social vulnerability. By using Atlantis, we are also looking into impacts on changing prey interactions. In terms of survey planning, given the northern shift in habitat, it is important to maintain the current dynamic acoustic survey footprint to ensure that all northern sardine subpopulation habitat is sampled. In terms of stock structure and transboundary management, as the northern subpopulation moves north, the southern subpopulation will become more important for California. And so it's important to continue the cooperative surveys with Mexico that were started in 2021, as that will allow us to evaluate the drivers of the distribution of the southern subpopulation and eventually collect enough data to develop a stock assessment. Our work has also shown that California fishing communities will be those most impacted by the projected shift. There is a high confidence across our 3 ecological models, as well as our 3 Earth system models that are represented here by the 3 different colored lines, that the sardine distribution will shift north. So here we show the center of distribution, or center of gravity, COG, of the sardine distribution by latitude. So that is increasing across all 3 of our ecological models. There is high confidence also related to this that the California contribution to total landings of northern sardine will decline, whereas the Pacific Northwest contribution will increase. If you're interested to learn more about our socioeconomic work, I suggest you check out the presentation by our economic postdoc, Felipe Quesada Escalona, in session 16. It shows how there are differences in the potential adaptive capacity of the CPS fleet by port and by fleet segment. And also, I encourage you to listen in to Isaac Kaplan talk in this session. In addition to the impacts of changing species distributions, managers and stakeholders in our region also want to understand how changing stock productivity will affect stock assessment and stock forecast performance, the effectiveness of management strategies, and the downstream impacts on ecosystems and socioeconomics, as well as impacts on food security and nutrition. In the next slide, I will provide a brief overview of how what Future Seas has learned in terms of changes in stock productivity is going to affect the first 4 points. So far, we have assessed how changes in recruitment affect stock assessment performance and management strategies, as well as ecosystem impacts and socioeconomic impacts. In the future, we plan to also assess how changes in adult growth, as determined by the individual-based model, as well as changes in natural mortality, are affecting stock assessment performance, as well as robustness of management strategies. If you're interested to learn more about the work on the impact of changes in recruitment, I encourage you to check out Robert Wildermuth's talk in session 15. He has developed a management strategy evaluation of harvest guidelines for sardine, as well as sardine stock assessment performance. And there are a range of recruitment scenarios, including climate-driven recruitment scenarios. Also check out Pierre's Zervan talk in session 15. He's going to provide an overview of the climate-informed ecosystem management strategy evaluation that uses Atlantis as an operating model so that we are able to assess ecosystem impacts as well as socioeconomic impacts under climate-driven recruitment scenarios and climate-driven shifts in species distributions. I'd like to end with a brief overview of what we learned so far. First, it's important to tailor your modeling framework to the specific context, and this means being aware of the management concerns and management priorities, as well as the data limitations in your region and the ecological and physical context. In terms of workflow, the first step is always to calibrate and fit your ecological or economic models as driven by the best historical environmental data to ensure that they can capture past dynamic. However, once you force those by projection, it's also important to go back and check that both the mean and the variability in the projected ecological or economic quantity of interest over the historical period is comparable to that was driven by the best historical environmental data. It is also important to capture uncertainty and to use ensembles to do that, and not only uncertainty across Earth system models, but also across ecological models. We found, for instance, that the relative influence on the center of gravity of sardine distribution of ecological uncertainty was larger than that of those than other sources of uncertainties by end of century, as you can see in the figure below. Finally, it's important to consider scenarios and socioeconomic futures for a heterogeneous fleet. This is work that we plan on focusing on in the future. Thank you for listening, and again, thank you to all the collaborators and co-PIs and postdocs that have worked on the Future Seas project.
We have just a couple minutes till we need to do the next talk. I'm trying to see if all our speakers are still here. If folks have questions for them that they didn't get to ask. Were there any questions? Kelly, do you want to ask yours? Okay, let me see if Jeroen's still here. Can Kelly ask you a question about your talk?
Excuse me?
Yeah.
Hey, thanks. Yeah, I thought your talk was very interesting. I'm curious, I feel like in my experience, no matter how universal one tries to make a coupler, models are always managing to obscure their inputs and outputs in a way that it's very hard to connect. I know you're very familiar with the eco-ocean model that you used in the example. Do you think that the process of getting things set up would be equally efficient, or maybe not equally efficient, but equally doable with a model that you were unfamiliar with?
Good question. In principle, yes. I mean, this framework just runs on the presumption that you just string command line applications together. So whatever the command line application can offer you, you can talk to. So it depends on the capabilities you wrap within what you can do with it. So there are no strings, there's no bounds, there's no limits, but it depends how the model is engineered.
All right, we'll go ahead and move into our next talk by Camilla Novagio.
Hi, thank you, Phoebe, for the introduction. So I am Camilla Novagio. I work at the Institute for Marine and Antarctic Studies, and today I will be talking about using ecological theory to evaluate and reduce uncertainty of a marine ecosystem model ensemble. No. Yeah. So you probably are sick of seeing this plot by now in this section, but they do show that global marine ecosystem models have been used to produce projection of climate impact on marine ecosystem and fisheries, and these projections have been included in policy report. However, the uncertainty around this projection, as Ryan pointed out this morning, is quite large, and it also is shown in these two maps of variability and agreement across fish meat models. To reduce the uncertainty of this projection, we need to validate our ecosystem model, but again, in this section, we've seen how difficult it is to validate these models. Typically, what we are doing in FishMIP is that we run models and we get some results. And at this point, we have two paths. We either analyze projections as mechanism, which is what we have been done so far, or we test the validity of this model. Testing the validity of these models requires some data against which we can compare our model output. We would go on this loop, keep improving our model until we have until our model meets some until we are happy with our model. They've been validated, and we run them again. The problem in validation of the global ecosystem model is that we are lacking data at the global scale, so it is quite difficult to get to the point in which we have information that we can compare with the model estimate, as it usually happens for the regional models. While we do not have much information at the global scale to validate our model, we have an understanding of how communities are structured, and we can use this understanding for model validation. Size spectrum theory in particular predicts that communities are characterized by a high number of small individuals and a lower number of big individuals, and that the slope of the relationship between the abundance and size approximate -1. This relationship is grounded in empirical evidence, and it is quite well studied. There are many books on this relationship, and it already informs our ecosystem models, which are based on trophic or size assumptions. Our ecosystem model can also produce size spectrum, community-level size spectrum, and slopes. We expect our ecosystem model to conform to theory and to also produce a size spectrum slope of -1, but we haven't tested this yet. We haven't used this information to validate our model. And this study represents a great opportunity for such testing. Here we specifically want to answer two key questions. What do global fish marine ecosystem model predict the size spectrum slope is? And how does the model slope relate to theory and to the available empirical information? We also want to know if we can use the size spectrum slope to constrain our model. To do so, we used model biomass for 6 size classes that are provided by 5 FishMIP ecosystem models that were able to output this information. We know that the normalized biomass size spectrum slope is about -1, as theory predicts, and we also use slope estimates from the Reef Life Survey global dataset. So we calculated the slope, we calculated the size spectrum and the slope of the size spectrum for each of the 5 marine ecosystem models that we are considering in this study. We used information from the recent past, so we considered the decade 2005 to 2014. And we were kind of glad to see that the global ecosystem model overall conformed to theoretical expectation, as they all produced a size spectrum slope of -1. There are, however, some variation between ecosystem models. However, at much smaller, a much finer spatial scale, at the 1 degree resolution, at the 1 degree grid cell, we can see that the size spectrum slopes shows variation and clear geographical patterns clear geographical patterns which differ substantially across models. It is worth noticing that some patterns do not only differ, but they are opposite. So if we look at both, we can see that there is a steep size spectrum slope at the pole, whereas if we look at ecotroph, we can see that the steeper size spectrum is along the coast of temperate regions. So there is a huge difference between these models. One other very important thing that we can see is that the DBPM projects a range of size spectrum slope that is quite high, with a minimum slope of -8, which is very unexpected. So this map raised a lot of questions. How do these maps compare to data? What is the acceptable range of slope that we can have in our ecosystem models? And how can we reduce the uncertainty of this ecosystem model, which is going to arise in future projection if the past looks so different? One of the approaches that the climate modeling community is using to evaluate and validate and weight ecosystem model is called emergent constraint. It is not been applied to the ecological systems, but it's quite interesting. So the plot on the right shows how the approach works, and the plot on the left is an application of this approach. So what we would do here is, first we find a statistically significant relationship between two model property, where these blue dots are the Earth system models. On the x-axis, we have an observable trend of variation in the historical period. For example, an historical trend in temperature. On the y-axis, we have a future change in a second property. For example, future precipitation changes. Then we would define the observed value. We would look at data and see what is the range of values that have been observed for the x-axis. And we use the intersection between the observed range and the emergent relationship to constrain our future projection. So we do like this approach, and we would really like to apply the same approach to our ecosystem models. In here, we wanted to relate the minimum slope, which we know is problematic for some models, to future percentage change in exploitable biomass between 10 grams and 100 kilograms. We plot our ecosystem models, and because we are working with emergent constraint and ecosystem model is quite difficult because we have a reduced set of models. It is challenging to find a statistically significant relationship. So we can't really call this an emergent constraint approach, but we've been inspired by this approach. And we've seen that when we define the range of observed values, which are defined using the Reef Life Survey data, a global yet patchy data set, we can clearly see that one of the models lays well outside this range, and that these models also project very steep percentage changes in exploitable biomass. So this model seems to be problematic. While this relationship can help us constrain, if we were applying an emergent constraint approach to this pattern, we would be able to reduce the uncertainty in future projection. It also allow us to understand what are the problems in our ecosystem models, to define what is happening and what are the issues. So based on this approach, we look at the raw data for the DBPM model, which is the problematic model. And we see that in open ocean grid cell, the one that show very steep slope, the raw data show this type of size spectrum relationship with a very steep slope. And this steep slope is due to nonlinear pattern in this relationship, but also to a point of inflection at very low size for grid cell in the open ocean, which is likely due to the limited amount of food. So if we correct for this, if we change some of the parameters in our model to correct for this pattern, we have an improvement and a size spectrum slope that approximate values that can be found within the range of observed values. So the expectation is that if we run the model with the improved set of parameters, this DBPM, the problematic one, will be moved to the top right corner together with the other model. And we would not only be able to use the emergent constraint to reduce uncertainty on future projection, but we will also be able, most importantly, be able to correct a pattern on the DBPM model. So this is a step forward in model improvement. So throughout this experiment, we've learned important lessons. The first one is that our ensemble overall conforms to theory, but the map of slopes show important issues, some of which we haven't yet solved. We used an emergent constraint-inspired method coupled with ecological theory to validate our ensemble and to guide model improvement. We would like to work more with emergent constraint for ecological models because we think they are very promising, but they are also very challenging because we are working in a very different setting from the physical world where this approach has been applied widely. We have limited and biased data, and we have higher complexity and a lack of understanding of some mechanisms that prevent us to explore meaningful relationship between two ecological properties. So to move the emergent constraint approach forward, we need more data as always, and a deeper understanding of how different mechanisms lead to ecological properties so that we can explore relationships that are grounded in theory, but also in well understood. And so that we can move from one emergent constraint, once we solve it, to the next one, to keep improving our model. We also need to explore relationships that we understand, but we have not yet considered in this context, because this is quite a new approach. Thank you.
All right, we have plenty of time for questions. There's one person behind Isaac, and then Isaac.
Hi, thanks. An interesting presentation. With the emergent constraint analysis, there's the statistical aspect, but there's also the understanding aspect. So you presented that there's this wide range of slopes, and you said that there's one model that is completely out. So, you could discard this model because it's wrong, and you have tried to, but I think you always have to have a basis for understanding why there's a change.
Yes, so this relationship is based on our understanding of how, it's based on our understanding. So if you start with a model that has a very steep slope in the history, that means that there are less large individuals in the community. And if temperature increases, and this can be due to lower transfer efficiency so that the big individual don't get enough food to survive, if you in the future reduce food and increase temperature as these scenarios indicate, then you'd have even less food for the big individual to survive. And that is why this relationship to us was meaningful to explore, and that is why we have this outlier model. Did I answer to your question or not? Half, not really.
Okay.
Thank you very much for a great talk, a very helpful talk. In the EcoPath world, there's a set of constraints that was proposed by Jason Link called the Prebell constraints, and it's very similar to what you're doing here, but it's looking at just as an example, the expectation of slope with respect to trophic level. Mm-hmm. I wonder if you've thought about testing that particular set of constraints and seeing...
Yes. Yes, the problem with this ecosystem model is that the output that we have are not that many, so we cannot really... So what the global ecosystem model output is the total consumer biomass, the consumer biomass divided by 6 size classes, the biomass of demersal and the biomass of pelagic. They don't output, for example, biomass by trophic level or many other information that would be very useful to explore the emergent constraint, other emergent constraint. By the way, I'm not calling this an emergent constraint because there is a lack of statistical relationship. It's just that by looking at this, we've found important patterns.
All right, thanks, Cami.
Hello? Hello? Okay, here we go. All right, thank you for coming back from the coffee break. So we are in session 8, advances in coupling socio-ecological models. And we've got 6 more talks left in the session this afternoon. And first up, we've got Isaac Kaplan. And the title of his talk is Forks in the Road, Models to Support Climate-Ready Ecosystem Management in the California Current. Take it away.
All right. Sounds good. And I'll ask my engineer to put the slides on the floor as well as the ceiling. Thank you. Thanks very much. So I'm Isaac Kaplan. I'm going to talk about forks in the road and some modeling that we've done and decisions we've made in the California Current, thinking about climate change. And I want to thank all of my co-authors, especially the co-authors who are online at odd times of the day, as well as those in the room. So if you were here earlier, you heard a really great presentation by Desiree Tommasi introducing one of our climate change projects, the Future Seas project in the California Current that looks at pelagic species. So you already know all about that. My favorite thing about the Future Seas project is really that it's a chance to integrate species distribution models with ocean projections and food web representations and trophic dynamics. It is really a chance to think about climate change and bringing those impacts down to the level of individual ports and communities. And of course, there's great talks and posters throughout the session and throughout the conference, and you may have seen those already. In the California Current, we also have a project focused on groundfish and climate change. This is what we call the GC5 project. It's also thinking about climate change impacts, in this case on groundfish species, their trophic dynamics, their fisheries, and the human communities that rely upon them. And one of the questions we're asking here in the GC5 project is really to think about whether our harvest policies, whether our threshold harvest control rules for groundfish are robust to climate change. And on the right here, you can see one of our typical threshold harvest control rules used for these species. So to ask this type of a question about climate change impacts on the California Current, I believe you need ecosystem tools to ask these ecosystem questions. The tool that we're using here is an Atlantis ecosystem model. It's been developed by Beth Fulton in Australia at CSIRO. And it's a 3-dimensional simulation model, a dynamic model that's projecting fishing impacts and other human activities, trophic dynamics and ecological relationships, and oceanography. In the California Current, we have implemented this for the US West Coast and then a portion of southern Canada and a portion of northern Mexico, and extending out to the extent of the exclusive economic zone. Okay, putting work aside for a second. Coming up in May, there's a fantastic 7-mountain tour around Bergen. So there's 7 mountains that ring the city. And so you should come back on the 28th if you don't live here and hit these 7 mountains. So the problem is that nobody holding my passport has ever done the 7 mountains. So I'm going to take you on a tour of 4 mountains today, and we're going to walk through the decisions that we make building these models as we move through these 4 points. Okay, so I'm going to take you up the mountain. We're going to go up to Mount Ulriken. We're going to hike up here, starting down in the city. And we're going to think about decisions at the top of that mountain, thinking about ocean downscaling. So you've already heard from Desiree Tommasi about the gold standard, as Tyler mentioned. The gold standard in climate projections is dynamical downscaling. In the California Current, we have an amazing set of work by Mercedes Potzobuel and colleagues, which is dynamical downscaling, looking at climate change impacts to both the physics and the biogeochemistry under different Earth system models. This is fantastic, and it's really the bedrock of all of the Future Seas modeling, the whole ensemble of models that Desiree Tommasi was just talking about. There's one trick, though, which is that the spatial extent of this model doesn't quite cover the spatial extent of Atlantis. And so the Atlantis has these tails that hang out beyond the downscaled, dynamical downscaled ROMS domain. So we're actually taking a simpler approach. We've made a decision to take a simpler approach, in particular using a delta method that superimposes anomalies from the Earth system model onto a high-resolution GLORIES reanalysis. And I know there's folks from GLORIES here at the conference. And so Owen Liu has devised this delta method, and it's really a pragmatic approach to fill in regions where we simply don't have the dynamical downscaling products. And there's a poster touching on that as well. So we made this decision to use a delta method for the downscaling, for the oceanography. We're going to hike along and hike over to where you see that ice cream cone at Mount Floyen. That's just up the hill here. and we're going to think about species distribution models. So species distribution modeling is clearly one of the fields that's really advanced dramatically since the last ECWO conference. There's an amazing set of work on this. Everyone's talking about it. And in the California Current, we are really lucky to have species distribution modeling for the recent past and long-term projections for pelagic species and for groundfish species. For pelagic species, we have contributions from Barb Muehling, who gave a keynote a couple days ago, and Nerea Lezama Ochoa and others from the Future Seas team. And then for the groundfish, we have SDM TMB projections, so that's the modeling framework. Owen Liu has been conducting those, and he already presented on that work in session 10. So we have these projections of pelagic species and groundfish through the year 2100. And that really the question and the decision point is, how do we treat those? How do we incorporate those into a model like Atlantis? And so the decision is essentially, do we let those species distribution models just define the fundamental niche or the broad habitat space, or do we actually use that to represent the realized niche or the actual, the true location that we model the fish to be in or the species to be in on any particular day or any particular simulation time step? For a variety of pragmatic reasons, we've made the decision to force Atlantis with the SDMs as a realized niche, really driving the model to precisely capture those interpretations from the SDMs, from the species distribution modeling. That was the decision. So we're going to go with the realized niche. We're going to hike over to Rundemannen, which is our third peak. And I just want to mention some of the decisions about the fishing fleets. So fishing fleet structure in ecosystem models very often, especially in Atlantis models, very often has involved just forced catch time series or forced fishing mortality time series. Those are really simple representations, and I think we're doing something better and making better decisions in this current round of work. In particular, we've really focused on the particular fishing locations of the major ports. So this is for the pelagic species fishing groups. The coastal pelagic species ports are shown here. Each port has a particular fishing radius. These vessels are quite constrained to stay near the coast because their product spoils very rapidly. And we're really mapping the footprints of these fleets on our ecosystem. And we can go one step further. So we can take the Atlantis projections of catch per port, and we can partition those further to catch per vessel cluster or per vessel behavioral type, or you might say métier. So for instance, we can think about catch that might be landed in a port but might be harvested by sardine opportunists, or by industrial squid vessels, or by more roving industrial vessels, larger vessels. So we can do that type of partitioning down to the vessel cluster level. And in a similar way for the groundfish fleets, in the GC5 project, Jamil Samhouri and Becca Selden and colleagues have really focused on trying to understand from logbook data where the fishing vessels actually fish, and the trawl vessels especially. And so we have very detailed footprints of where those fleets operate on the map and on the model domain. And then also we can further partition the catch to vessel behavioral type or métier. So that was that decision. I'm going to hike over to results. These are sort of preliminary results, but I'll take you there anyway. These are 50-year projections thinking about the world under an IPSL projection, RCP 8.5. We're going to take a coast-wide view first. This is biomass. So this is the biomass at the end of 50 years relative to a run to a simulation with a 50-year projection with climate change and species shifts relative to a 50-year projection with no species shifts and no climate, essentially just constant climate. We see winners and losers. We see, in particular, some of the winning species, the species that increase in biomass, are the pelagics, so some of our herring, sardines, small pelagic fish. We also see some losers, especially some of the groundfish, and I'll just point out the flatfish groups in particular. We see 4 flatfish groups declining in abundance relative to the base case in terms of biomass. The catch results are shown here. Again, this is catch at the end of 50 years with climate change relative to a base case with no climate change and no species shifts. Again, winners and losers. To some extent, these results mirror what we see in the biomass projections. So for instance, we see increases in catch of some of the pelagic fish in red on the right. And we see some declines as well for species like arrowtooth flounder that was also declining in biomass. But the catch results don't exactly mirror the biomass results. So for instance, we only see one of our flatfish groups, not 4 of them, declining in abundance. And that's because the catch results are dictated not just by changes in biomass, but also changes in species distribution and how that overlaps with the fleet operating areas. Fishers and communities really are less interested in that coast-wide view. They're much more interested in what's happening in their own backyard. And so we can take this down to the level of individual ports. So here are our trawl fleets arranged from north to south. We can look at catch relative to the base scenario. And we see, for instance, increases in catch in some of the northern ports, generally the mid-latitude to higher latitude northern ports, for 4 of our key groundfish target species. We see declines, however, in a couple of our northernmost ports. And the point there is that the species distribution models are predicting that some of these target species are actually moving, not so much moving north, but they're moving deeper. They're moving offshore and out of the range of some of these fishing fleets, or at least the historical range, and also some of the regulatory bounds on those fleets. For the small pelagics, we see a similar story, changes in catch by port. Again, here the ports are arrayed from north to south for the CPS or coastal pelagic species fleets. We see increases in sardine catch for some of the northern ports, and that's been predicted by other models, as Desiree Tommasi pointed out. We also see declines in some of the anchovy catch for particular ports, and that's the blue bars there relative to a base case. And anchovy is really not one of our major target species. It's sort of a niche species for some fleets some of the time. But we can just focus in on anchovy for a second and look at what that change in anchovy catch means for a particular vessel cluster. This is just looking at anchovy catch relative to the base case, and just pointing out that for one particular Pacific Northwest fleet, that's the fleet or the métier or the vessel cluster that essentially feels that decline in anchovy the most due to both their distribution, their abundance, and their overlap with that fleet. So we can get down to the vessel cluster level. So in summary, I think we're refining these models to really think about oceanography, species shifts, and bringing in other data as well, including new diet information. We're really thinking about ports and communities and getting down to the level that the fishermen care about in their backyard. We're moving from just biophysical representations to representations that think about what communities feel and what ports feel. And we're also beginning to test management strategies and to evaluate which management strategies are robust to these changes and this changing climate. And there's some great presentations in session 15 by Pierre-Yves Heneghan and Robert Wildermuth on that theme. So with that, thanks so much to Pisces, ICES, and IMR, including for a workshop on Sunday on these themes. Thanks, Tyler.
All right, thanks, Isaac. We have time for one quick question, if we have any. I see Cheryl.
Thank you. Thank you for the tour of the peaks. I guess my question is, does Atlantis use nutrients or NPP or some kind of lower trophic level forcing? And if so, how did you apply the delta method to that? And how do you think that uncertainty might propagate through to these management? This is something I lose sleep over.
Absolutely.
Yeah, and I would love your opinion.
Absolutely. Yeah, so this is a great question. So we're using the nutrient phytoplankton zooplankton model within Atlantis. We've talked about using an alternative approach, which would force Atlantis with nutrients and phytoplankton and zooplankton from a separate model, say from an Earth system model. We haven't done that yet. So I think that, yeah, that's an alternative approach. But yeah, we have dynamics within Atlantis evolving for those components. Yep.
Great. Thanks, Isaac. All right, next up we have Olivier Maury with the title Assessing the Time of Emergence of Global Oceanic Ecosystems Using Ensemble Climate to Fish Simulations. Go ahead, Olivier.
All right, thank you, Tyler. Today I'm going to talk about assessing the time of emergence of global oceanic ecosystems using ensemble climate-to-fish simulations, as you just said. I'm afraid I'm sorry, so again, a global talk in a regional session, so thank you for accepting our hacking, and we had no other home, actually. Okay, so this work was conducted in the framework of the Tree Atlas project, which is an EU project led by Noel Kinley-Syke, who is here in the room. And I would like first to acknowledge my co-authors, Nicolas Barrier and Mathieu Languen from Eddy and the Marbec Laboratory, as well as Roland Seferian and Irène Santana in the CNRM in Toulouse. So I think you have seen this graph already in this session. As you know, of course, climate change is altering global ocean ecosystems at an increasing pace. And the FishMIP ensemble simulation has highlighted that we can expect something like between 10 to 25% of biomass decrease in the SSP 585 at the end of the century. This is pushing ecosystems towards a radically different states, completely novel states with no analog in the past. So in this context, the time of emergence could be useful to assess this novelty. So if you look at a variable here, a climate variable or other variables through time here, the time of emergence, the TOE, is the time at which the climate change signal, here in red, exceeds the magnitude of the noise induced by natural variability, which is here in blue. So the TOE is here, when the signal exceeds the noise. Okay? Many studies have assessed TOEs of physical variables. However, this has not yet been attempted for marine ecosystems. And here we will try to do it, and we assess when changes in marine ecosystems emerge in simulations from the APECOSM Global Marine Ecosystem Model, forced by an ensemble of physical and biochemical simulations from the CNRM climate model for 2 SSP scenarios, long sentence. APECOSM is actually a mechanistic model of marine ecosystem which is based on first principles. It expresses processes such as dynamic energy budget, bioenergetics, physiology, size structure, opportunistic predation, 3D movements, cooling at the individual level. Then in the model, we upscale individual dynamics to derive Eulerian population dynamics, which in turn are upscaled to get the 4D, X, Y, Z, and size structure generic community dynamics using a trade-based approach. So here in this talk, we use an archetypical tree communities model configuration where we explicit a epipelagic community, the dynamics of migratory mesopelagic community, and resident mesopelagic community. Following the FishMIP protocol, the climate change simulation protocol, APECOSM was forced by SSP-driven climate model outputs, such as temperature, marine currents, turbulent mixing, phytoplankton and zooplankton biomass, particulate organic carbon, oxygen, and light. And that allows us to simulate the dynamics of the tree communities in the model and look at their changes through time within the SSP scenarios. So what you can see here is that the communities display different rates of decrease. A epipelagic community decreases faster, minus 22% at the end of the century, while migratory community decreases slower and reaches minus 3.7% at the end of the century. Resident mesopelagics have sort of intermediate decreasing rate here. And the total that we look at in FishMIP, the TCB, total consumer biomass, is actually reflecting a sort of average of these three weighted by their respective abundance. That's the decrease, the change rates of the TCB in space, and you see, I won't detail it, you see that it is quite heterogeneous, especially with areas, especially in the gyres, where biomasses are expected to increase, while in most other areas, it is expected to decrease. To compute the times of emergence, the TOEs, we follow the SEMIP6 simulation framework. We first spin up both our ESM and marine ecosystem models. Then we run a 500 years PI control simulation here that allows us to estimate the PI is pre-industrial, in case you don't know this term, which allows us to estimate the natural variability, namely the mean, mu, and the standard deviation, sigma, there. Then we run both historical and scenario simulations for 2 SSPs, and we do that for 5 members. So it's 79 computing days on our cluster HPC. So it's a huge amount of simulations, actually. It looks simple like this, but it's quite heavy. And we don't have ERN's framework to do it. And then we calculate the average of the 5 members over the entire simulation period. That's the black line that you see here. And to extract the climate change signal in every model cell, we use a 30 years Gaussian filtering of the 5 members average that you can see here in black. And it allows to remove all the interannual and decadal variability that is remaining in the signal. From that, we can compute TOAs as the time at which the climate change signal exceeds the envelope and does not come back into it. So let's have a look at the TOEs for marine ecosystems in the SSP 585. So here you see the bars on the left axis here. The bars are the percentage of cells, of model cells, that are emerging per decade. The lines are the total percentage of cells that are emerging through time here, and that's the right axis here. So in blue, you see temperature that is emerging quite early. It starts emerging in the 70s. It peaks in the 2010s, and 85% have emerged before present, 85% here. And at the end of the century, almost 100% will have emerged. For mesozooplankton, it is emerging later. It starts emerging in the 90s. It peaks in the 2040s. 20% have emerged before present, and about 63% has emerged at the end of the century. And finally, for epipelagic organisms in green, they emerge later than temperature, but earlier than zooplankton. They start emerging in the 90s. There is a steep peak in the 2020s, that you can see here. 27%, so more than 1/4 of the ocean, has already emerged now, and about 62% will emerge at the end of the century. Okay, so the question is now, what determines TOEs for surface temperature? So you see here TOEs. The color is from the 60s in dark pink here until the 2080s in dark green here. And you see that temperature is emerging early almost everywhere. It's almost everywhere pink, but in the Central and Western Pacific Ocean. You also see that TOEs mirror closely the signal-to-noise ratio, as could be expected. And these are huge, actually, in this. Namely, it means that the signal is very strong compared to the noise for temperature. Signal is responsible for the first order of TOE pattern. We have done a correlation pattern analysis, which shows that signal drives the first order of TOEs, the warming signal here, and noise modulates the second order slightly. For instance, in the Central Pacific, you can see the signature of the strong variability, which is due to the thermocline movement when Rossby waves, ENSO Rossby waves, are going through the ocean. What determines TOEs for low trophic levels and high trophic levels? So you see here the TOEs for mesozooplankton. 37% of the surface in gray will not emerge at the end of the century, and the rest, of course, 63% emerge. TOEs again mirror signal-to-noise ratios. There is a strong noise near the coast, but quite a weak noise elsewhere. It's very blue here. And highly contrasted signal. You can see here blue and red areas for the signal. But in the end, it is again signal rather than noise that is dominating signal-to-noise ratios, but noise plays a stronger role than for temperature. And for epipelagic community, you have signal patterns that are similar to mesozooplankton. They drive the first order of signal-to-noise ratios. There is a strong and heterogeneous noise, which modulates the signal-to-noise ratios. High trophic levels emerge earlier than low trophic levels, and we believe that might be due to a trophic amplification due to temperature here. TOEs for other HTL communities, that's the three mesopelagic, migratory, and epipelagic communities here. What we can say is that despite smaller signal than for epipelagics, the smaller noise makes mesopelagics and migratory emerge earlier than the epipelagics. So that was a surprise for us. They start emerging in the 80s, so almost 10 years before epipelagics. So there is also a substantial geographic difference between communities, but I won't go through that now. The size structure of TOEs for high trophic level communities, what you see here on the left, you see actually the color is the percentage of impacted ocean, emerging cells, as a function of here, and size of organism here, from 0 to 2 meters. What we see here is that small sizes are emerging earlier and stronger than large sizes for epipelagic organisms. This is because noise, you can see noise here as a function of length of organisms, so noise in blue for epipelagic is actually increasing quicker than signal with size, which lead to an amplification of flow frequency, the decadal variability for large sizes. And we are currently investigating this amplification of the decadal signal in the large sizes. For both resident and migratory organisms, we see a sort of U-shape with small and large sizes emerging earlier and stronger. And this is because while noise is increasing steadily, we observe that signal is actually increasing later. It is first steady and then it increases, but sharper than for noise. And that leads to this sort of U-shaped pattern that you see here. Okay, the conclusion now. That's the first time that TOEs are calculated for global marine ecosystems. And our method based on coupled ESM, MEM ensembles allows a good approximation of both noise and signal. TOEs could provide an interesting metrics for assessing the emergence of novel ecosystem states in fish MIP ensembles, for instance. And what we have seen as well is that biological variables do not have the same TOE patterns than physical variables. Surface temperature is emerging 30 years earlier than LTLs and 20 years earlier than HTLs. There is a strong sensitivity of HTL TOEs to the size and community. And you haven't seen that, but that's the other scenario that we have studied. While temperature signal emerges everywhere in both SSPs, the surface where biological variables emerge would be divided by more than 3 by the end of the century in SSP 1 to 6. And finally, we have pointed the fact also that while it is not really important for temperature, noise has a critical importance for biological variables TOEs. Even if it is second order, it is modulating quite strongly the patterns, meaning that the propagation of interannual to decadal variability in marine ecosystem models should be investigated further more than what we have been doing to date. So I use this opportunity in this perspective to point this paper that we just published, and which is actually digging into details into the mechanisms that are conveying the interannual variability and zoo variability in the ecosystem in the apicosm model. I thank you for your attention, and I'm ready for.
Thanks, Olivier. I think we've got time for a question, if we have any.
Yeah, the 500-year pre-industrial simulation to establish the noise level is probably an obstacle for a lot of people repeating this type of simulation. Is there any way to shortcut that, or is it?
Why would it be an obstacle?
Because it requires an awful lot of computer power.
Ah, that's for sure.
Yeah. Because yeah. I mean, have you done? I guess what the question is: have you done experiments that kind of ascertains, how long you actually need to run the pre-industrial run to capture a particular amount of the noise signal?
No, we could do that quite easily using the 500 years and
subsampling,
shortening it artificially. Yeah,
okay.
We did not.
Yeah.
Yeah.
All right, thank you, Olivier.
Thank you.
All right, next up we've got Joe Caracappa with, did I say that right? Yeah, okay. Application and development of the Northeast U.S. Atlantis model to address climate change impacts on a regional socio-ecological system. Go ahead, Joe.
Well, that was a very ambitious title. So I'm more talking about what we're trying to get to and more of an advert and a solicitation for a recommendation. So this session's been fantastic. We all have seen this a million times this week, but the Northwest Atlantic in particular is warming faster than global averages, and we have a wide range of species in the Northeast U.S. shelf that have a variety of vulnerability to potential climate impacts. And on top of that, we also have a very diverse array of coastal communities ranging across our region that are experiencing different degrees of commercial engagement in fishing and reliance on fishing. So the vulnerability of each individual community can be quite different. And so the question that I've seen here a lot today and that we've been thinking a lot in general is how to bridge some of these global concepts into actual community level decision making while actually maintaining the complexity of the ecosystem and some of the nuances we see in these scenarios. And so we're taking, similar to what Isaac said, an Atlantis approach, and so some of this might be a little redundant, but it's an end-to-end ecosystem model, so we're simulating biogeochemistry through human socioeconomics. The advantage of Atlantis is that we do have an international framework that we can work off of and collaborate with, but each individual model is calibrated to individual regions based on regional data and with regional questions in mind. So its modular framework makes it very flexible to address a wide range of questions and to try to scale the trade-off between the scope of a model and how specific the question can be. But it's also a multi-species model where we have spatio-temporal processes, full food web resolution, and it's mechanistic at its core, meaning that we can derive a lot of emergent processes from it. In general, this is kind of how an Atlantis model works in modular form. You have your drivers of the system feeding into a biological submodel, and then that leads into a management and socioeconomic loop. We are pretty much in this space right now. It's kind of a beast to work with, and it's a little finicky, but it's worked in other regions, and we're kind of aspiring to complete that management cycle in the simulation. And so broadly, our goals for the Northeast Atlantis model, Northeast US Atlantis model, are to create a set of baseline climate projections under different climate change scenarios where we can then use these near-term projections to facilitate the discussion of what different fisheries adaptations for the region might be on a community level, and then try to bridge those into the language of fishery socioeconomic and community vulnerability. And so our approach for this is a little different than what Isaac was presenting, but we're focusing mostly on a hindcast model right now to try to establish that the processes working in Atlantis are actually functional and that we're getting the right sensitivity that we're expecting. So we've been working a lot with the physical drivers, the biological drivers of the system, and making sure that when we do project that we're getting reasonable results. And then from there, we moved to a validation phase where we're using sensitivity analysis on these processes and trying to stress test the model before we even project using climate scenarios to try to make sure that, like I said, that those processes are working, while also validating against whatever assessment and survey data we have for the region. And then we're hoping in the coming months to the next year to start integrating much of what has been talked about today as far as climate projections downscaled from a regional model while implementing a variety of fisheries adaptation scenarios. And so we've gotten around this portion of it ready, but I'll be sharing a little bit more about that in a moment. The Northeast model in particular extends from Cape Hatteras through the Gulf of Maine. We have 89 different functional groups over a variety of taxa, and we have 30 spatial polygons that we operate over. Our hindcast model, we are forcing it with a downscaled Glorious 12 reanalysis for temperature, salinity, and circulation. We're actually forcing primary production in the model using a regional satellite-based ocean color product, where we actually, on a daily level, overwrite Atlantis's internal NPZ dynamics with this product, so driving the food web and instilling the seasonality of the system onto the model. We're using all the commercial and recreational landings data that we have to actually force fisheries as a time series in the hindcast. And we're trying to calibrate as much as we can, like I said, with trawl and assessment data. And so one big question that we had that was kind of outstanding was Atlantis is a box model. All boxes are kind of uniform within, and we wanted to make sure that we were actually capturing the spatial and seasonal structure of these environmental drivers that we were forcing the model with. And so we did some work that got published last year to try to evaluate that. And despite the coarse resolution, we were able to capture everything but the gradients within boxes, obviously, but we still maintain the seasonal structure and the overall regional spatial patterns and the environmental and lower trophic drivers. We also did some time series deconstruction work to try to make sure that the seasonality of the dynamic zooplankton were reacting as expected to the phytoplankton. And while the timing of the seasonality was a little phased off, we still got a succession from phytoplankton to small, medium, and large zooplankton. And so we were pleasantly surprised by that. As of now, we're working on a kind of a broad sensitivity analysis to fishing pressure just in the hindcast model. So we're projecting kind of business as usual, but at several different fishing levels to try to evaluate what the landscape of the mean catch in the model is with regards to exploitation rate and how far you could push the system before individual species start to crash. And we're particularly interested in where in Atlantis there are refugia from fishing that are not really well documented, and trying to make sure that those are kind of taken care of before we start projecting, because that's going to be a major issue for us. And so just sharing some of our thoughts with regards to where we're planning on going in the future. We have in the Northeast, we have a MOM6 cobalt product that's using a downscaled version of GFDL's 2.6 model, which is a much higher resolution than we've previously been able to use, and it's able to resolve much of the coastal bathymetry and circulation, and we're hoping that that'll resolve some of the issues that have happened in the past with position of coastal currents and in general. And we're planning on forcing the entire lower trophic as well as nutrient pools in Atlantis, so we have that as a complete one-way coupling. A major issue conceptually is in Atlantis. Isaac touched on this a bit, but how do we tackle the question of temperature dependency in Atlantis? We basically have to choose from a set of tools that are already kind of pre-built in there or come up with our own. And the ones that are in there right now and have been used historically are direct methods, where either we modulate metabolic response for individual species to temperature, we define their thermal niches, or we do temperature-dependent recruitment that's scaled in a variety of functional forms. And these all have kind of pros and cons, and most of the cons are related to just kind of time investment and the finickiness of it and how long it takes to calibrate. But we also have some indirect methods now where we can use external products and force directly, either through scaling like phytoplankton growth and keeping it dynamic, but just shifting it to match what some external product is defining, or as Isaac was showing, species distribution model forcing. And so we've had a lot of conversations about this week, and I think we have a game plan, but I won't spoil it. The other main question that we've been tackling is with regards to fleet definitions and fisheries parameterization. And we have this kind of spectrum in Atlantis from fixed catch through dynamic effort. And I only know of one or two Atlantis models that have gotten this truly dynamic fisheries module working, where fleets respond in real time to current conditions. So usually we're left to prescribe what the fleets have to do during a projection, and that can come with its own biases. We're moving towards this kind of space where we have either a fixed time series of effort, and instead of just catch or fishing mortality, but it has its own difficulties in implementing. So we have the opportunity now to really spend some time on redefining the fleet structure in the Northeast US model. And so we decided to take a communities at sea approach, where we define fleets by not just by their home port, but also by vessel size and gear type, to try to really bridge that global to local gap. And due to some unknown reason, the Northeast US model runs very fast, so we have the opportunity to test a large degree of scenarios ranging from changing target species, fishing footprints, and seasonality to actually like really encompass the space and try to test the sensitivity of Atlantis in general to these different factors. And we're hoping to work this into some form of decision support at the community level, but I've been trying to solicit some feedback on that throughout the week. Building off of some work that was done by Gavin Fay a couple of years ago with an older version of the Northeast model, he was able to link it with the Northeast Regional Input-Output Model, which is a socioeconomic model that takes fishing effort from Atlantis and catch composition and a few other metrics and combines them on the county level on land to produce socioeconomic indicators that we can tie directly to those community-based fleets. And so with the addition of different climate scenarios in the future and the higher degree of flexibility we have in these fisheries adaptation scenarios, we're hoping to provide a much more kind of cumulative, comprehensive way of assessing these adaptations. And then lastly, the Northeast Fishery Science Center conducts a State of the Ecosystem report every year where we try to evaluate the current conditions of the different fishing communities we have in regards to environmental justice and social vulnerability. And we're hoping to use Atlantis to provide not necessarily a recommendation, but a contextualization of where each of these ports are currently and where they might be with regards to the same indicators to get stakeholders thinking more about kind of how things might change from a format that they're used to seeing. And so with that, I thank you all for, this has been a great session, and I'm happy to hear any feedback, and special thanks to all my collaborators in the Atlantis community.
Thanks, Joe. We've got 3 or 4 minutes for questions, so. Yeah, Kim.
Thank you. I'm not sure if you can answer, but I just looked at that kind of social impact, the one before. Yeah, is emissions from the fleet something that you consider in this? Do you know?
So the version that this was done in has been updated since the economic model. So I'm not actually sure whether emissions are in there, but that would be
Yeah, I was also
possible.
thinking about it in a previous talk. It's something to include. I think it would be interesting to see.
I think it is ignored often, so it would be helpful to have that in there.
Any other questions for Joe?
Hi, Joe. Thanks for a great talk. I was wondering if you could just tell us more about what types of refugia from fishing you were talking about when you mentioned that in the fishing tests.
So it comes down a bit to how the selectivity curves in the model are defined and what the fleets have access to, both spatially, with age, and over time. Atlantis has a lot of options. In our model, we found that certain age classes were kind of exempt from fishing because they were supposed to be in estuaries for mostly juveniles, but the definition that we had for when they enter the fishery might not have been correct.
One more question, yeah, Anna.
Thank you, Joseph. That's great. Great work, obviously. My question is about how do you bring everything together? So whether you, in Atlantis, do you have the ability to then combine? So you assess sort of, ecosystem climate sensitivities, and then you showed economic impacts, and then you're talking about social vulnerability. And then for decision making, do you then just give those to the practitioner to weigh, or
I don't think that would be the most efficient way to do it. I think what we were intending, it's funding dependent, but creating some sort of online tool that has all these scenarios in the background that lets them pick what would be most appropriate for their situation and then work through this scenario space that we've constructed on the background. But thank you.
Thanks, Joe. So we have one more talk in this session, and we have Sophia Wassermann, who's going to be talking about cannibalism and other trophic dynamics of Pacific hake in a changing ocean.
All right, thank you very much. This talk is going to be a lot more in the weeds than some of the other talks have been. It's not a global assessment, it's a very specific question. And this is part of my postdoctoral work at the University of Washington in collaboration with a bunch of wonderful people from the Northwest Fisheries Science Center. And essentially, I'm talking about ways to answer a question that's important for climate-informed and ecosystem-based management, where the complexity of an ecosystem model isn't necessary, but where we think that trophic interactions are important for answering questions around the population dynamics of a target species. And my target species is Pacific hake. So they're a really important commercial species in the California current ecosystem, and they're a very abundant predator, which also means that they're an abundant prey species for other fish and marine mammals. They're considered generalist predators despite having a diet that's largely consisting of euphausids, but their pacivory increases with age. And there hasn't been a big study of hake diets since the 1990s, but at that time, cannibalism was estimated at 30% of their diets, reaching a maximum of 75% in 1999. I'm using the Seattle model, which we've heard a little bit about already. It is the climate-enhanced, age-based model with temperature-specific trophic linkages and energetics. And it is a multi-species statistical catch-at-age assessment. So it's a single-species models linked through predation mortality. I'm using the R-Seattle package, which I wanted to mention because it makes it very easy to run. And it's all on GitHub, maintained by UW grad student Grant Adams, who just presented in the other session. And Kirsten earlier referred to this model as the basis of a climate-enhanced multi-species assessment for some species in the southeastern Bering Sea. So it is being used to supplement assessments. And it differs from a single species model in the way that natural mortality is comprised. So the numbers at age of each species for this year is a function of the number of age of each species for the previous year times this total mortality, which is broken down into M1, residual mortality, M2, predation mortality from the predators included within the model, and then the fishing mortality. And the predation mortality is derived from the suitability coefficient, which comes from prey, and age preference, which comes from the stomach contents of your species, then the estimate of prey biomass, and an estimate of annual consumption, which comes from temperature-dependent consumption and consumption with body size. So this is highly reliant on good diet data. And this plot is the occurrence of cannibalism in collected hake stomachs from the Hake Acoustic Trawl Survey off the west coast of the US. And this is from 2005 to 2019. And the colors here, purple is stomachs that did not contain hake, and the green is color as stomachs that did contain hake. And you may be thinking, I can't even see the green. And that's because out of 4,000 samples collected from that time period, only 16 contained hake. So this estimate of 30% cannibalism didn't seem to hold true, which was a little worrying. But when we looked earlier on using data contained within the California Current Trophic Database, we found higher records of cannibalism. And so that's in the 1990s, corresponding with the studies that took place around that time. And you may still be thinking that doesn't look like 30%, and that's because this is the frequency of occurrence of cannibalism, but we're doing the proportion by weight. And that's ends up being a much higher proportion because when cannibalism occurs, it takes up most of the space in the stomach. So this is the diet proportion by weight for the entire time series averaged. So the predator hake ages are on the x-axis, and then the prey hake ages are in the different colors. So the majority of predation is on age 1 hake, but there is predation on hake up to age 5. And I want to point out that this proportion reaches approximately 15%. So when we plug that in to the Seattle model, this is what we see. When the mean proportion is included in Seattle, it increases the estimate of mortality. And the model therefore estimates higher biomass than number of recruits to fit the survey indices and the catch data. And so this is spawning stock biomass, total biomass, and number of recruits for the single species assessment for the Seattle model with just with no trophic interactions. So that's in blue. And then for the Seattle model with cannibalism in purple. And you'll see that there is a slight increase between the single species assessment and the single species Seattle model, and that's due to an increase in the estimated M1s, that's the residual mortality, over the assessment value. And then another increase with cannibalism, which is a result both from the increased mortality from predation, but also an increase in estimated residual mortality. But you see, so that's responsible mostly for this change in scale, but you see some increases, especially here, and also earlier on in the time series for all of these factors. And we think that that is a result of the age structure at the time. So another way to look at comparing these is to look on the left at the numbers at age. So that's on the top is for the model with cannibalism, and on the bottom is for the stock assessment. And then also at the total mortality on the right. So we can see much higher mortality for the age 1 hake. And also this change over time in the estimated mortality and also in the age composition. And so that raises the question for me of what could we do for time-varying diet? And it would be great to fit a fully time-varying diet curve across this, but I don't think I can really interpolate what's happening between the two main periods that we have for diet data. So the graph on the left here is the one you've already seen, and on the right is what I've done just to try to think through this question. And at the top is the diet proportion that I showed you before with a maximum of around 15%. In the middle is the diet data for 1988 to 1999, which reaches up to 80%. And then at the bottom is the period for 2005 to 2019, which is this much lower diet proportion that we saw from the Hake Acoustic Trawl Survey. And I fit those to Seattle just for the time periods represented by the data. And I really don't want to imply that this represents time-varying diet, but it's just to look at interactions between potential changes in cannibalism and in the age structure of the population. So in purple now is the diet proportion for all of the years. In blue is the most recent years from 2005. And then in green are these earlier years of the data. And it shows what we would expect with much higher biomass in recruitment estimated for the model when cannibalism is higher, again amplified by the age structure for that period. And so the question I now have is what leads to this variability in cannibalism? This is probably really difficult to see, and it's also far from a spatial temporal analysis that other people have presented in this conference, so I'm a little embarrassed to talk about it. But I just wanted to take a look at the location and the time of sampling to get a sense, some sense of why this change has occurred. And so if you squint, you can kind of see that maybe in some of the early years, like 91, and here in the 90s, the sampling is occurring, where you see cannibalism is occurring a little bit further south and maybe a little bit later in the year. And cannibalism relies on the overlap between the older and the younger, the larger and the smaller individuals. So it's possible that cannibalism is occurring outside of the sampling period or further offshore. But cannibalism also depends on what other food sources are available. So in thinking about why cannibalism has changed over time, I wanted to mention this work that Beth Phillips and others have done, looking at the overlap between hake and euphausids in the California current. And they found a higher co-occurrence with euphausids during cooler conditions than in the neutral or warm conditions, which suggests a climate link not only for consumption rates, as is included already in Seattle, but perhaps for prey availability and choice. So putting this all together, the hake diet data suggests that there has been a change in the amount of cannibalism. If hake can be assumed to be cannibalistic by opportunity and not by preference, then that suggests that the older and younger hake aren't overlapping as much recently, or that a different or preferred prey source is more available. And I can't really answer these questions without doing a more thorough analysis of the recent diet data. But taken on average, cannibalism does have an impact on the estimated size of the population by increasing our estimates for for natural mortality, especially for age 1 hake. And this effect is magnified by the age structure, as when there are more larger individuals in the population, there's more predation pressure on the youngest of the hake. And cannibalism has been shown previously to have an effect and potentially destabilize established population cycles. And so, I mean, potentially just sort of thinking about this time series of recruitment within hake, Maybe that could be partially responsible for the recruitment spike in 1999, following multiple big cohorts in the population in the late 1980s. So lastly, cannibalism only represents a very small proportion of the total predation mortality on hake. So moving forward, we're interested in including more predators within this model. And this is a list of the most voracious hake predators included in the California Current Trophic Database, ordered by the frequency of hake predation relative to their occurrence within the database. And you'll see hake fall about halfway down that list. And so the species that we were thinking about looking at next is California sea lion. And I wanted to talk about this because I'm interested also in how this, maybe looking at an additional data source of hake predation, can show us something about how the population dynamics of hake have changed over time before I've actually plugged it into Seattle. And so the graph on the left is the frequency of hake predation in sea lion scat samples. And I think it's really interesting to note these slightly higher rates of hake predation that fall off towards the mid and late 2000s, which somewhat corroborates the trend in cannibalism that we see we have missing diet data for, which suggests that maybe there were more small hake available in the 1990s to both larger hake and to sea lions. And the sea lion data set also provide information about when hake are found in the sea lion stomachs, at least in Southern California where these data were collected. So the graph on the right is the monthly sampling of stomachs, and the highest number of records is for June, or for July, but there is some predation, at other times of the year. And I think that's a really great side effect of including multiple species in your model. You can get a picture of what happens outside in the system outside of your survey period. So just in summary, including these kinds of predator-prey interactions depends on high-quality diet data that can be harnessed through models like Seattle to inform both past and present population dynamics. And additionally, these models can provide insights into how natural mortality varies with age or with time for incorporation into single species stock assessments, even without being used as a full multi-species assessment. And increases in estimates of biomass and recruitment with cannibalism only represent part of the predation mortality present in the system. So including more predators will only provide a better picture of overall natural mortality. And with that, I'd like to thank everyone who has contributed so far to catching me up to doing this work, many of whom are at this conference, and for the Northwest Fisheries Science Center for the funding.
Thank you, Sophia. We've got time for a couple questions.
Hi, I like that talk a lot. So coming from the outside, I'm not a biologist at all, and listening to all these talks this afternoon, it strikes me that one of the things we're interested in is the change with time and climate change and temperature. And yet you have this great data set where you could look at what the temperature was in all these different seasons and what was going on. I mean, there was a lot of stuff happening back when that one pool was out there, particularly to seals, so those are your predators, but maybe to everybody. So maybe relating your results to just temperature would be interesting.
Absolutely. I think that a wider look into what's happened with the hake diets and also consumption of hake by other predators in relation to temperature is really important. And certainly the euphalcid or krill connection is strongly linked to temperature. And as the main source of food for hake, especially for young ones, that will have an impact on the rest of their consumption. And I haven't had a chance to look deeply enough into those diet data to make any or to draw any conclusions about how temperature plays a role, but I think inevitably it does.
Do we have a final question? Okay, I think everyone's bandwidth has reached its capacity. Thank you, Sophia. So I'd like to invite the co-conveners up just to officially close this session, so come on up. So we'd like to thank all of the contributions, all the early career ocean professionals, all the posters, all the speakers today, our invited speaker, Ryan Heneghan. So please join me in thanking everyone. And I'll pass it over for my co-conveners for any final remarks.
I just really thank all of the contributors to the session. It's really interesting to see the step change in advancements of these integrated models and approaches and analysis of sources of uncertainty since the last ECWO. So it's super exciting, and thank you for your contributions, and thank you for coming to hear the talks.
Yeah, I was inspired by you guys, and you didn't let me down, so thank you.
Yeah, thanks to all. I'll pass it to you, actually.
Oh, no, I was just going to say it's really cool to see how everyone's using kind of complementary approaches or similar approaches to answer different questions. So I thought that was really cool. So thanks, everyone, for your contributions.